Hello everyone, very good evening and welcoming you again to this amazing platform of PW NEET English, and again your zoology mentor, myself Biswajit. Today in this session we are going to complete the human physiology from biology. Yes, we'll be covering every aspect of those chapters today. Total 6 chapters we are going to cover. Will be one by one, the chapters will be covered with practicing few of the important questions, and obviously for every chapter one or two very much important mechanism is there. We have to complete them. After completion of the chapter, we have to cover each and every disorders related to every human physiological aspect. So hello, welcome Sudeep, welcome Priya. So how you all are doing? Let me know in the chat section before starting the class. So Sudeep, where from you are, and you will be giving for the 2026 NEET exam, or you are preparing for next year? Similarly, what about Priya? What about you? Where from you are watching the video, and currently you are in which class? Total time the class will be running till 12 o'clock. We have to complete whole human physiology, complete human physiology. Duration will be 8 hours. Okay, Sudeep, so you are from West Bengal. That's great. So, কেমন আছো Sudeep? Hope everything is fine. Okay, so at the rate from future, you are from Kashmir. Oh, great place. That's great. So, you people are studying in which class right now? Class 11th, class 12th, or in dropper? Preparing for NEET 2026. Okay. So, let's start with the first chapter of today's session, that is breathing and exchange of gases, and this is the first chapter given in our human physiology section. If you check your NCERT, okay, quite laser. Hello, good evening. Okay, so from future, I don't know what's your name. You are studying in class 12. That's great. Okay, so when is your board exam, or is it over already? You are preparing for 2027. That's great. Sudeep, okay, from where in West Bengal? You are watching from which place, which district of West Bengal, Sudeep? Okay, you are studying as a dropper. That's great. Okay, so let's start the first chapter, breathing and exchange of gases. And when we listen this terminology, breathing, that's quite synonymous with another term of biology, that's respiration. So here very briefly I have discussed what is the basic difference between breathing and respiration. So basically breathing is the process of the mechanical process, physiomechanical process of inhalation and exhalation. There is no utilization of direct ATP or energy, whereas respiration means it's a kind of biochemical events happening inside our cells specifically, and the sub events under respiration, if we consider the aerobic mode of respiration, then they are glycolysis, Krebs cycle, electron transport chain, and obviously it's a chemical reaction happening inside our cell and breaking down glucose into simpler molecules. So it's a catabolic kind of reaction. I'm talking about respiration, and in a better manner you can call as intracellular respiration. So every day we are eating food material, we are taking nutrients as carbohydrates, proteins, fats, vitamins and minerals, and we know that these food materials, specifically carbs, protein and lipids, they are rich with energy. Now in order to get those energy, we have to break down them with the help of oxygen, either directly or indirectly. So that's the process of taking oxygen and supplying oxygen, taking from atmosphere and supplying that oxygen to each and every cell to break down or to oxidize the glucose. There is the simpler process of whole and soul breathing and exchange of gases. So here diagram You can observe. So the basic process of intaking air and exhaling air, this is the process of breathing, or you can call it pulmonary ventilation. Then with the help of our respiratory passage, the air will be reaching into the respiratory bags present inside our lungs called alveoli. Now can you please mention in the comment section that how many alveoli are present into our body? How many alveoli are present into our body if you consider both the lungs? Okay, so Sudeep is watching from South 24 Parganas. Okay, that's great. Okay, quite ledger. Your name is Subir Biswas. That's great. That's great. So I had a question here for you people. So who can tell me that if you consider both the lungs, how many alveoli are present inside us, continuously doing exchange of oxygen and carbon dioxide? Can you write down in the comment section? I'm waiting for your answer. So the first step is the process of exchange of gases or breathing. You can call these as pulmonary ventilation. Then there will be exchange of gases in between alveolar air and our blood. Exchange of gases in between alveolar air and blood. Now it's the duty of our blood to circulate, to carry, to transport that oxygen rich blood and to deliver to our organs. Near to the organ, again there gonna be another exchange of gases in between blood and organ. Here blood is releasing oxygen into the tissue, into the cells of organs, and those cells of organs are releasing carbon dioxide, and that making the oxygenated blood into deoxygenated. So organs are getting oxygenated blood, and the blood which is carrying carbon dioxide, that is becoming deoxygenated, and via heart again the deoxygenated blood will be approaching towards the lung. Again oxygenation will take place, and continuously the process is going inside our body. Okay, so let's talk about that diversity we are finding among different groups of animals. The animal kingdom is highly diversified, and different animals have different habitats. Few are staying inside water, few are staying in outside water, few are terrestrial animal, few are aquatic animal, few are living in desert. So depending upon their habitat and body design, different animals have different respiratory structure. So let's talk about that. If you talk about Porifera, Coelenterata, Ctenophora, platyhelminths and aschelminths, all these primitive animals, they do not have any special respiratory structure. They will be exchanging the gases with the help of their body surface by the process of simple diffusion, right? Because when they are producing carbon dioxide, inside their body there is more concentration of carbon dioxide, and outside their body there is less concentration of carbon dioxide. So carbon dioxide will be coming out or going out from body, and oxygen will be coming in. So that is the process of simple diffusion, and they prefer their body surface or general body surface. Now if we talk about annelids, now which animals will be coming inside annelids? They are earthworms, leeches. They have their moist cuticle to perform the exchange of gases. If we talk about arthropod, the largest phylum in the animal kingdom, insects among arthropods, they prefer tracheal respiration. They will be exchanging the gases with the help of trachea and spiracles. Yes, good evening, NEET MCQ shorts, that's great. Then if we talk about arachnida, now who are arachnids under arthropods? Scorpions, spiders, they are considered as arachnids. a butterfly, cockroach, ants, they are considered as insects. So spider and scorpions, they have book lungs kind of structure. With the help of these book lungs, they will be performing respiration. For crustaceans like crab, shrimp, prawn, they are aquatic in nature, so obviously they have gills for respiration. And living fossil called king crab or horseshoe crab, they have book gills, a special structure modified for exchange of gases. Now coming into the phylum Mollusca, means snails and octopus. So molluscs, particularly snails, they have feather-like gills for respiration. Mollusca, they have feather-like gills, also we call them Ctenidia. For Echinodermata, starfish, they have dermal bronchi. Also they can respire with the help of water vascular system. For the group of animals called hemichordata, as because they are also exclusively marine or aquatic, they have gills for respiration. Who are hemichordates? Balanoglossus, right? Then it's our phylum, that is chordata. Under chordata, urochordates, cephalochordates, cyclostomes and fishes, means pieces, they will be respiring through their, with the help of their gills. And amphibia, reptilia, aves and mammals, generally we respire or we exchange our gases with the help of our lungs. Now we got different names for different modes of respiration. If you are respiring through your lungs, then that mode is called as pulmonary respiration. Respiration through lung is pulmonary respiration. Respiration through gills for fishes, respiration through gills is called branchial respiration. Respiration through skin, like in earthworm, in frogs, is called cutaneous respiration. For cockroach and insects, they are respiring through trachea, that is called tracheal mode of respiration. So you need to remember these terminologies, right? Pulmonary respiration, cutaneous respiration, branchial respiration and tracheal respiration. So if I ask you a question, the respiration through gills, is called pulmonary, cutaneous, branchial or tracheal respiration. You can answer. You can write down your answer in the comment box. What do you call the respiration through gills? Is it pulmonary, cutaneous, branchial or tracheal respiration? Yes, Muhammad Afla, very good evening. So I got a question for you all, that the respiration mode through gills is called as pulmonary respiration or cutaneous respiration or branchial respiration or tracheal respiration, and the answer is here, it's branchial respiration. Yes, option C. Now we'll be moving forward to our own respiratory system. Now we'll be talking about respiratory system of human being. Okay. So we know that we have one pair of lungs acting as the biological machine for exchanging the gases. So a respiratory passage starts with one pair of nostril. or external nair. Then inside our nose we have nasal passage, so let's talk about first up to throat region. So our respiratory system starts with our external nostril, external nostril. Then inside our nose we have nasal chamber. Inside our nose we have nasal chamber, and remember that nasal chamber or inside our nose, it's always moist. We have to maintain moisture because air conditioning starts from our nose itself. Then here at the end region of our nose, at the end region of our nasal chamber or nasal passage, we have internal nair or internal nostril. So from external nair to internal nair, this zone is called as our nose region, and near to internal nair we have a highly coiled space that is responsible for perceiving the smell. So that's called olfactory epithelium. Okay, now where this internal nair is ending, after that this throat region we have a common passage of air and water and food, that is called pharynx. After internal nair we have pharynx region. Pharynx region is a common passage of our oral cavity as well as nasal region. So that's why we say the pharynx is common passage of air, water and food, and pharynx is having three zones. Towards the nose we have nasopharynx. Towards the mouth we have oropharynx. And towards the larynx we have laryngopharynx. So these are the three important zones of our larynx. Now from larynx there is division of two zones. One tube will be going towards food passage. So towards the towards our dorsal side we have our esophagus, and towards the front side we have our trachea or food pipe. So the bifurcation of these two tubes started from the pharynx region only. Now the initial part of our trachea is modified into a special structure for sound production. We call it as larynx. So larynx is nothing but initial modified version of trachea only, and inside the larynx we'll be finding some vocal cords. That's why larynx is also called as voice box. Inside larynx, how many vocal cords are there? We have 4 vocal cords. Inside larynx, we have 4 vocal cords. That's why larynx is called as voice box. Okay. So that was the anatomy of our respiratory system up to our throat region. Now The opening of our larynx is very much crucial for us. The opening of our larynx is called glottis. The opening of our larynx is called glottis, and the opening of our esophagus, that is called as gullet. That is called as gullet. So basically it's like this. If you check your throat, after your pharynx, that is divided into two portions. One is called as trachea or windpipe, and behind the trachea you have your esophagus or food pipe. So this one is your food pipe and this one is your windpipe. The opening of your windpipe is called as glottis. The opening of your food pipe is called as gullet. Now when you are inhaling there is no problem, but when you are eating food or drinking water, then if by chance, by mistake, if food particles enters into your windpipe, then what will happen? That will ultimately block your respiratory passage and you are gonna die. So that's why the glottis is always enveloped or protected or closed during the time of our swallowing, when we are drinking or eating of food material, with the help of flap of elastic cartilage called as epiglottis. A special kind of elastic cartilaginous flap which is protecting the opening of your windpipe, that is called glottis. And remember, the initial part of your windpipe is called as larynx. And you all know that throughout the windpipe you will be finding some incomplete C-ring structure. Throughout the windpipe you will be finding some incomplete C-ring cartilaginous structure. This C-ring cartilage, they are present throughout the length of your trachea or windpipe, and that's helping your respiratory passage from becoming collapsed. Because whenever food will be passing through your esophagus, the esophagus will undergo what? The esophagus will undergo peristalsis movement, continuous contraction and relaxation movement, and that may exert, that may create extra force or extra pressure on your windpipe, and that may collapse or block your windpipes. Now thank God that Almighty has already given us this C-shaped incomplete ring structure. So whenever this peristalsis is happening inside your esophagus, that extra pressure cannot disturb the air column present inside your trachea or windpipe. Okay. Yes, now we were talking about up to our throat region only, right? Now gradually the trachea will enter into the lungs. Now gradually the trachea will be entering into both the lungs. We have two lungs, left lung and right lung, right? And the place from where the trachea is getting splitted into bronchi or primary bronchi, that is written as T5, is very much important for us, near to fifth thoracic vertebrae. Near to fifth thoracic vertebrae, the trachea is getting splitted into right and left primary bronchi. Now let's talk about this tracheal bifurcation and the pipelines present inside our each and every lungs. So what I just said, that the trachea is getting bifurcated near to what? Near to T5, that is fifth thoracic vertebral bone. Okay. Now this bifurcated part, these are entering into your lungs, and we will be calling them as 1 degree bronchi, means primary bronchi. Now further, this primary bronchi, when it's entering into your lungs, will be subdivided into a few groups. And this further branches of primary bronchi is called as secondary bronchi. Further, this secondary bronchi will get subdivided inside your lungs. The subdivision of secondary bronchi is called as 3 degree bronchi or tertiary bronchi. So, altogether primary, secondary and tertiary bronchi is part of our bronchial tree. So, bronchi is divided first primary bronchi, then secondary bronchi, then tertiary bronchi. Now, this cartilaginous rings or this cartilaginous plates will be continuing throughout the primary, secondary and tertiary bronchi. This cartilaginous rings or plates will be continuing throughout the primary, secondary and tertiary bronchi. Now suddenly this tertiary bronchi will get narrowed down. Now suddenly this tertiary bronchi will get narrowed down to form further narrower channels, and we call that this is the narrowest part or narrowest tubes present inside our lungs. So these narrow channels starting from your tertiary bronchi, we call them as initial bronchioles. Initial means at the beginning, initial bronchioles. And these cartilaginous plates can be observed even into these initial bronchioles. Even these cartilaginous plates can be observed into these initial bronchioles. Now, upon further subdivision, these initial bronchioles will be terminated by forming many more bronchiole structure, and the termination part or the end part of bronchioles, we call them, the end part of bronchioles we call them terminal bronchioles. Remember, these terminal bronchioles are not containing cartilaginous rings or cartilaginous plates. And after terminal bronchiole, what is present? After terminal bronchioles, we will find some respiratory bronchioles, means the last part, and this respiratory bronchioles will be giving rise alveolar duct and ultimately some balloon-like structure where ultimately respiration or gaseous exchange, external respiration or gaseous exchange will be taking place, and this balloon-like miniature structures are called as alveolus in singular and alveoli in plural. So what after terminal bronchiole? After terminal bronchiole we have respiratory bronchioles, then alveolar ducts, then we have alveoli. So this is all about the respiratory tree present into our lungs, and remember there are nearby 300 million altogether. For both the lungs we have 300 million alveoli, and for single lung we have 150 millions of alveoli present inside. For one lung we have 150 million alveoli, for two lungs we have 300 million alveoli. Now if I draw the passage of our air from the beginning, the starting is from external nostril. from external nostril to nasal chamber or nasal passage, from nasal chamber to internal nostril, from internal nostril to pharynx, pharynx to larynx via glottis, from larynx to trachea, trachea to bronchi, trachea to bronchi. Here we have to remember we have primary bronchi, secondary bronchi and tertiary bronchi. I have written is as 1 degree, 2 degree and 3 degree. Now bronchi will be giving rise to from the narrowest tube of a respiratory passage called as initial bronchioles. Initial bronchioles. Initial bronchioles will be subdivided into terminal bronchioles. Terminal bronchioles. Now you need to remember this. From trachea to initial bronchiole, from trachea to initial bronchiole, we have cartilaginous rings or cartilaginous plates are present. And here is the end of one story, means the transportation of air, the story of transportation of air from external nostril to terminal bronchiole, and this passage is called as conducting part. This passage is called as conducting part of air, because nowhere starting from external nostril to terminal bronchioles we could see exchange of oxygen and carbon dioxide. And question is asked that what are the function of conducting part? Then if gaseous exchange is not happening, then when air is traveling through the pipes, what's happening really inside? So the function of conducting part is to act as AC. AC means air conditioning of air. Now what do you mean by air conditioning? What do you mean by air conditioning? Match the temperature or to regulate the temperature of air, to regulate moisture of air. So that means air conditioning of air. See, whatever air you are inhaling, either that is dry or that is moist, either that is having high temperature than your body or that is having low temperature than your body. But whatever air, whenever you are inhaling air and it's reaching to your alveoli for gaseous exchange, you need to give it a perfect temperature, a matched temperature with your body. That means your body temperature is 37 degree, and if your room temperature is 25 degrees Celsius temperature and you are inhaling air with 25 degree, then it's the duty of your conducting part to increase the temperature from 25 to 37, and ultimately that will be reaching to your alveoli. So regulate the temperature, regulate the moisture, that is, these two are the main function of your conducting zone, that is air conditioning. And one more important, if you are inhaling dust particles, so these dust particles or bacteria, they will be filtered but with the help of the special kind of tissue present through your conducting part, that's a kind of ciliated epithelium, right? So with the help of your ciliated glandular epithelium, ciliated glandular epithelium present into your conducting zone, the inhaled air is filtered, means any dirty things, any dust particles are cleared now. So these three are the functions of your conducting part. Now what after terminal bronchioles? Now after terminal bronchioles we have respiratory bronchioles, Respiratory bronchioles will be subdivided into alveolar ducts, and alveolar ducts will be ended with the help of those balloons called as alveoli. Now here is the starting of another story. Here what will be happening throughout this respiratory bronchioles, alveolar ducts and alveoli, there will be exchange of gases. There will be exchange of gases. That's why from respiratory bronchiole to alveoli, this passage, the ending passage of your respiratory tree is called as exchange zone, exchange or respiratory zone, exchange or respiratory zone. So that was most of the things about our respiratory passage present inside our lungs. Yes, alveolar sacs, as because it's not mentioned in NCERT, right? So basically what happens, these alveolar ducts will give rise alveolar sacs. This alveolar ducts will give rise alveolar sacs. At the ending of each alveolar sacs, you will be getting alveoli. Okay. So these are the things we were discussing right now, and this is the diagram given in our NCERT book. Already we have discussed all these things, and now few more things are there to discuss with you. Now ultimately all these alveoli together forming our lungs, right? Ultimately all these alveoli are forming our lungs, and we know that this is our left lung and this is our right lung. Now can you tell me which one of them is larger and which one of them is smaller? If we compare between left lung and right lung, which is larger, which is smaller? Can you write down in the comment section? In between right lung and left lung, which is larger, which is smaller? Right lung is larger, exactly. Right lung is larger, and compared to right lung, left lung is smaller, and you know why? Because of the presence of or occupancy of the apex of your heart. Because of the occupancy of the apex of your heart is little bit tilted towards the left, and that's the reason it's occupying or it's creating a concave plane directly inside the left lung. So that's why left lung is compromising with its volumetric area, and that is the reason you'll be finding that the left lung is divided into only two lobes. Each lung is having further parts, we call them lobes. The left lung is divided into two lobes and right lung is divided into three lobes. So right lung is having superior lobe, middle lobe and inferior lobe, but left lung is having only two lobes, superior lobe and inferior lobe, divided by this kind of groove like structure called as fissure. Now you can check that the common fissure, if both the lung is having this oblique fissures, but this right lung is having an extra horizontal fissure. That is the reason there are three divisions of your right lung but two divisions of your left lung. So that's why we call that right lung is trilobed but left lung is bilobed. Right lung has three division, left lung has two division, and beneath the lungs we have a muscular membranous structure which is separating out our abdominal cavity from our thoracic cavity, and the muscular flap or muscular membrane that is called as diaphragm. And remember one more important thing about diaphragm. Yes, remember one more thing, important thing about diaphragm. Diaphragm is one of the exclusive feature of mammals. Means in fishes you won't be finding any diaphragm. In amphibians there is no diaphragm. In reptiles except crocodile there is no diaphragm. So diaphragm can be considered, the presence of diaphragm, which is basically a muscular membrane which is separating our thoracic cavity and abdominal cavity, a special characteristic feature of mammalia. Remember this. Okay. Now the last thing about our lungs, the covering of our lungs. That's because whenever we are performing inhalation and exhalation movement, there is chance the friction will take place, and that may damage your minute alveolar structure present inside your lungs. That's why Almighty has given two important coverings. Our lung, each lung is covered by two layers, and they are called as pleura. The covering of lung is called as pleura. Covering of lung is called as pleura. And remember, the pleura is having two parts. The outer pleura is called parietal pleura, and the inner pleura is called visceral pleura. And question is asked that in between parietal pleura and visceral pleura, which is present towards our body cavity, which is present towards your lung. So it's very easy that if this is your lung, if this is your lung, if this is the parietal pleura and this is the visceral pleura, then obviously this is the side of, this is the body cavity. The parietal pleura is towards your body cavity and the visceral pleura is towards your lungs. And the space in between two pleura, the space in between the pleura is called as intrapleural space, and that is filled with interpleural fluid. The space in between parietal and visceral pleura is filled with special kind of colorless fluid called as interpleural fluid, and that's helping the lungs to maintain the friction, to maintain the surface area or surface tension, and also this interpleural fluid is acting as shock absorber for our lungs or protector of our lungs while you are inhaling and exhaling. Yes, pleural fluid or interpleural fluid. So that was all about our human respiratory system and their anatomical structure in a superficial manner. Now you know that as because our lungs are very delicate structure, they are well protected inside our chest cavity, we call it thoracic cavity. Now thoracic cavity is a very sophisticated structure or airtight chamber. Remember this term. Our thoracic cavity inside which our lungs are present, it's an airtight chamber. Now what is the importance of airtight chamber? What is the importance of airtight chamber? That we cannot change the volume of lungs directly. We cannot change the volume of lungs directly. That's why we have to change the volume of thoracic cavity first. Then based upon the volume change of thoracic cavity, our lung volume will also be changing. So the relationship is that the volume of our thoracic cavity is directly proportional to volume of our lungs. That means your lungs are well protected and kept inside your thoracic cavity. If you increase the volume of your thoracic cavity, lung will also increase its volume. If you decrease the volume of thoracic cavity, lung will also decrease the volume. That's why remember this relationship between in between lungs and thoracic cavity. Volume of thoracic cavity is directly proportional to the volume of lungs. Why it's so? Because lungs don't have their own muscle. Our lungs are not muscular in nature. So if there is no muscle, we don't have direct capability to contract or relax it. That's why when we are inhaling, we are actually contracting some muscles of thoracic cavity, and when we are exhaling, we are relaxing the muscles of thoracic cavity, and that's maintaining the volume of lungs, that is volume up or volume down. When you are inhaling, we have to increase the volume of our lung, and when we are exhaling, we have to decrease the volume of our lung, and that is actually maintained by your thoracic cavity and the associated muscles present into it. Okay, you can check the channel PW NEET English, right? And there you will be finding the links of various batches. At the last, you can get the link of WhatsApp channel PW. The WhatsApp channel is called as PW NEET English WhatsApp channel. So after every lecture, we will be uploading the notes over there, or else you can explore the PW NEET English complete zoology batch by me, that is NEET English, Zoology NEET English by Biswajit Sir in PW app. From there also you can download the notes, right? Okay. So remember the airtight chamber thoracic cavity, and inside that we have lungs which do not have own muscle. That's why there is a direct relationship between thoracic cavity and the volume of lungs. So what are the different components of your thoracic cavity? What are the different components of our thoracic cavity? This is very much important. See, at the front part here, the mid-ventral part of our chest, we have a sternum bone. We have a sternum bone, and from sternum bone to our vertebral bones, from sternum bone to vertebral bones, continuously we have some flat bones called as rib bones. They are laterally protecting your thoracic cage or thoracic cavity. Sternum, where it's present, the mid-ventral region of your chest. Okay, ribs are present the lateral side of your thoracic cage, and at the dorsal side or back side you have your vertebral column. You have your vertebral column, and the space in between the ribs, that is called as intercostal space or space intercostalis, and inside that intercostal space you got some muscles called intercostal muscles. And when you are contracting or relaxing those muscles, that's helping you to inhale or exhale. The movement of your chest is due to the movement of these intercostal muscles, and as well at the posterior side of your thoracic cavity towards downwards, you have this muscular flap called as diaphragm. So it's basically together the muscles of diaphragm and intercostal muscles making you or helping you to increase or decrease your volume of thoracic cavity. And if volume of thoracic cavity is changing, it will be influencing the volume of your lungs. Okay, so that's why it can be observed that your lungs or our lungs are well protected inside this bony and muscular structure, right? Okay, so now I will be talking about a little bit more about those muscles. So if you are asked what are the muscles related to your thoracic cavity, what are the muscles of your thoracic cavity, then they are intercostal muscles. What do you mean by intercostal muscle? When you are studying your ribs, right, when you are talking about our when we are talking about our ribs, the space in between the ribs are occupied by some muscles. We call them intercostal muscles. And what about second kind of muscle? That is the radial muscle or phrenic muscle. Radial muscle or phrenic muscles of diaphragm. So these two kinds of muscles are related to our breathing movement, and if you get inside these intercostal muscles, there are two kinds of intercostal muscles are there. External intercostal muscle, EICM, and internal intercostal muscles. Okay. Now for our syllabus, we only have to talk about external intercostal muscle and radial muscle of diaphragm, because we need to study only about normal breathing, normal inhalation and normal exhalation. So during the time of normal inhalation, okay, during the time of normal inhalation, we have to contract EICM, external intercostal muscle, and the radial muscles of diaphragm, and normal exhalation we have to do the opposite phenomena, that is relaxation of EICM and relaxation of radial muscles of diaphragm. But if you want to perform forceful breathing, then only you will be needing help of IICM, that is internal intercostal muscle. For forceful breathing, you will be needing help of IICM, and along with that, associated to your abdomen region, we have some abdominal muscle. So that's not given in our NCERT. That is out of our NCERT. Forceful breathing, but NCERT is mentioning that normal breathing requires this and this muscle, but if you want to alter, if you want to do forceful breathing, you can use some accessory muscles, and these are these accessory muscles. They are internal intercostal muscle and abdominal muscles. Yes, Anshika, you wanted to tell something? Okay. Now, according to our NCERT, we have to study about the 5 steps of human respiration, and they are chronologically they are here. First is breathing or pulmonary ventilation. Breathing or pulmonary ventilation. You are breathing in or you are breathing out. That means that is divided further 2 sub steps. When you are inhaling, that is called inspiration. When you are exhaling, that is called expiration. Why we are calling this as pulmonary ventilation? Because air is going in your lungs and air is going out of your lungs. That's why the process is also called as pulmonary ventilation. Okay, thank you. Thank you, Subir. Thank you very much. Okay. Now when your lung has already taken the air inside, the air will be reaching into your balloons, that means into your alveoli, and alveoli is having very thin membranous structure which is facilitating the diffusion of gases in between blood and alveolar air. So the second phase is the method of diffusion, and here diffusion of oxygen and carbon dioxide will be in between alveolar membrane and blood. The very third step, now while this oxygen has already entered into your blood, making the deoxygenated blood into oxygenated one, it's the duty of blood to transport the oxygen to each and every tissue of your body via your heart, obviously. So that's a third step, transportation of gases by blood. And fourth step is like this, again another diffusion gonna take place, and that's in between blood and the tissue, where tissue are crying as hypoxia, we need to do respiration, please supply us oxygen and please take this carbon dioxide. So again another set of diffusion is gonna taking place in between the blood and tissue of your organs. So that's why the second slot of diffusion, the fourth step, diffusion between blood and tissue, and obviously the last step is utilization of oxygen by cell to catabolize the glucose molecules and to break it down in the form of oxygen and water and to give us energy. And that process is also can be called as intracellular respiration. We don't have to talk about intracellular respiration in this chapter. We only have to study about the process of exchange of gases, the structure involved, and how our blood is helping us to transport oxygen and carbon dioxide inside as a transportation vehicle of our body. Okay. So now the next mechanism is the mechanism of inhalation and exhalation. So let's talk about that, the process of inhalation and exhalation. Now it's a very conceptual thing. The chapter is standing upon the process that air always move from high pressure zone to air always moves from high pressure zone to low pressure zone. Air always moves from high pressure zone to low pressure zone. Okay. Now, who can tell me in the comment section what is the normal atmospheric pressure? Is there anybody who knows what is the normal atmospheric pressure? Can you please write down in the comment section what is the normal atmospheric pressure? Shubir, 159 millimeter mercury pressure is atmospheric pressure of oxygen. But what about the air? Air is mixture of oxygen, carbon dioxide, nitrogen, and a few other gases as well, right? So can you please tell me what is the atmospheric pressure of air, not oxygen? You are absolutely perfect. 159 mercury pressure is atmospheric pressure of oxygen, is the partial pressure of oxygen in atmosphere. I am asking about the pressure of air. See, air pressure or atmospheric pressure is nearby 760 millimeter mercury pressure. Now, if you are not breathing, if this is your lung, and if you are not breathing, the pressure inside your lungs gonna be the same. 760, no, it's 760, Rajesh, it's 760 millimeter mercury pressure, atmospheric pressure at sea level, right? So air pressure is 760 and your lung pressure is also 760. Then you are not inhaling or exhaling. Now what you need to do? Now you need to inhale air. That means if you want to inhale air, air will be moving from atmosphere to your body, and we know the rule, air always moves from high pressure zone to low pressure zone. And if you want to inhale, the air pressure is 760 and you have to create low pressure into your lung. You have to create pressure of 758. You have to create a pressure of 758 millimeter mercury pressure into your lung during the time of inhalation, right? So that will create a negative pressure into your lung and a positive pressure into air. During the time of inhalation, listen it very carefully. air pressure is always 760. Now you have to create low pressure or negative pressure into your lung. That's why you have to increase the volume of your thoracic cavity. When you are increasing the volume of your thoracic cavity, the pressure will fall. If you increase the volume of your thoracic cavity, the pressure will fall. Here you can recall the physics, the law of physics with biology. According to the Boyle's law, volume is having volume is inversely proportional to pressure. So if you increase the pressure, volume will decrease. If you decrease the pressure, volume will increase. So while you are inhaling, you are increasing the volume of your lung. So pressure will decrease and the air will come inside your lungs during the time of inhalation. Now what about the time of exhalation? During the time of exhalation, you have to decrease the volume of your lung. So while you are decreasing the volume of your lung, the pressure will increase from 758 to 762. Now the atmospheric pressure is 760. So what will happen? You are going to expire the air. So air will move from your lung to atmosphere and that will cause exhalation. Okay, so now let's talk about the process of inspiration. So during the time of inspiration, we have to create negative pressure into our lungs. What do you mean by negative pressure? Low pressure in compared to the atmosphere. So during the time of inhalation, the pressure of atmosphere is 760 and the pressure inside your lungs are 758. And how to achieve that? By contracting the diaphragm and by contracting the external intercostal muscle. So simultaneous contraction of diaphragm and external intercostal muscle, that will increase the volume of your lungs, and that will increase the volume of your lung by anterior-posterior axis. Yes, when your diaphragm is contracted, diaphragm is flattened. When your diaphragm is contracted, diaphragm is flattened, and that will increase the volume into anterior posterior axis of your lung. And when your external intercostal muscle is getting contracted, that will increase the volume into dorsoventral axis of your body, and ultimately the volume is increased, and that will cause sucking of air from or movement of air from atmosphere to your lungs, and inspiration or inhalation is completed. Now this is a time for expiration, and you have to create positive pressure into your lung. That means your pressure of lung should be higher than your atmosphere. Now atmospheric pressure is 760 always. You have to create 762 millimeter mercury pressure into your lungs. So how to achieve that? You have to do opposite phenomena. You have to relax your diaphragm, you have to relax your external intercostal muscle, and simultaneous relaxation of both of these structure will move your rib towards the inwards and downwards, and diaphragm will become dome shaped again, and that will decrease the volume but increase the pressure. When intrapulmonary pressure increases, that will throw out the air outside of your body and causing exhalation and expiration. So yes, very important point said by Subir. Whenever we are performing inspiration, that's basically active process. That's an active process, and if you are performing expiration, that's kind of passive process. That means muscles are getting contracted, and muscle contraction needs ATP, and whenever ATP is involved, that is called active process. So inspiration is active process and expiration is passive process, because during the time of expiration, muscles are getting relaxed. Muscle contractions are always active process, muscle relaxations are always passive process. And if you are spending money, means for biologically money is ATP, if you are spending money behind some work, then obviously you will get faster result. That's why the timing for inspiration, one inspiration is 2 seconds, and for normal expiration is 3 seconds, right? So overall within 5 seconds you are performing one inspiration and one expiration. Rajesh, actually I cannot explain in Hindi because the channel is for pure English only. I'm sorry for that. You want Hindi explanation, but this channel is purely for English explanation only. This channel is created for the sole purpose, those who cannot understand Hindi or English, we are with batches for students who will be who were like more towards English, right, who love to understand the things or explain the things into English only. So I'm sorry in that aspect. Okay. So what I was telling, that inspiration needs 2 seconds and expiration needs 3 seconds, right? So within 5 seconds you are performing one inspiration and one expiration, and overall in a single minute you are performing 12 to 16 time of breathing. So that's your breathing rate, 12 to 16 time per minute. Within a minute you are inhaling or exhaling how many times? 12 time inhalation and 12 time exhalation. So these are the diagrams given in your NCERT. Now what kind of question can be asked? See, this is the diagram of inhalation, this is the diagram of exhalation. So you have to remember the axis. During the time of inhalation, in which axis a lung volume is getting increased. So the axis is, this axis is called anterior dorsoventral axis, and this axis is called anterior posterior axis. So in both the axes the volume will be increasing during the time of inhalation, and during the time of exhalation for both the dorsoventral axis and anterior posterior axis the volume is gonna be decreased. Okay. Now we will talk about the most important part of the structure of this chapter, and repetitive wise, like if you observe the PYQs, most of the questions from this particular chapter either you are going to get from this topic, that is respiratory volumes and capacities, or you are getting question from this chapter from the topic called oxygen and hemoglobin dissociation curve. So these two are the most important topics of this chapter. So let's complete this one, respiratory volumes and capacities. Now how to measure that? For that we need simple machine called spirometer, right? So you have to blow air inside that machine, and that machine can measure your lung volumes. So basically we have to talk about 4 lung volumes, right? So spirometer, the volume of air involved in breathing, or for clinical assessment purpose we can measure these lung volumes. And if you are going with spirometer, a very important thing, it cannot measure the residual volume. We know about 4 lung volumes. TV means tidal volume, ERV means expiratory reserve volume, IRV means inspiratory reserve volume, and RV means residual volume. Remember, spirometer can measure your tidal volume, expiratory reserve volume, inspiratory reserve volume. Spirometer cannot measure residual volume. For that, in higher classes, right, like suppose if you are getting admission in any government medical college or any medical college, for the first year of your degree you will be learning what are the different methods by using which you can measure the residual volume. But for us we need to know about what is tidal volume. If you are performing normal breathing, then you can inhale 500 ml of air and you can exhale 500 ml of air. So that's simply your tidal volume, right? So 500 ml is tidal volume per breathing, and if you want to convert this tidal volume into a minute, then 6000 to 8000 ml is your tidal volume per minute. If you are asked what is tidal volume per breathing, then you have to answer it's 500 ml, and if you are asked what is the tidal volume per minute, then you have to multiply 500 with your breathing rate and you are gonna get 600 to 6000 to 8000 ml. So if 12 is your breathing rate, if 12 is your breathing rate, then 12 multiplied by 500, you are gonna get a 6000 ml per minute tidal volume. Now what is expiratory reserve volume? Expiratory reserve volume means after a normal exhalation, if you are performing forceful exhalation, if you want to become Ramdev Baba, then forcefully another 1000 ml of air can be exhaled out. That is called expiratory reserve volume, and that is sometimes also can be called as supplementary volume. So in exam, by chance you get this word supplementary volume, don't think that's not given in NCERT. The another name of expiratory reserve volume is called as supplementary volume. Okay. Now IRV means inspiratory reserve volume. After a normal inhalation, means you have taken 500 ml of tidal volume, now you want to inhale forcefully. Now upon a tidal volume, after a normal tidal inhalation, by giving force, you can inhale extra 300 ml of air, that is called inspiratory reserve volume, and do not forget the another name of this, that is complementary volume. And residual volume, you all know that if you suddenly you become Ramdev Baba, and you want to throw out all the air present into your lungs. Now after practicing the highest possible way to exhale all the air, still you are not able to exhale this 1200 ml of air from your lungs. So that is the residual volume. After performing the most forcible exhalation, the air cannot be exhaled, always remain into our lungs. That is called 1200 ml or residual volume. Okay. Now I think most of you all know this trick to remember. If you multiply 2 with tidal volume, you will be getting expiratory reserve volume. As because tidal volume is 500, it's multiplied by 2, you are gonna get 1000, that is expiratory reserve volume. If you multiply by 3 expiratory reserve volume, you are gonna get 3000 ml, that is inspiratory reserve volume. Okay. Now we got inside our NCERT, we got a name of another volume, that is called dead space volume. Now what is dead space volume? What do you mean by dead space? The zone of your respiratory passage, if you eliminate the exchange zone or the respiratory zone, right? So if we observe our respiratory system, the conducting part always contains 150 ml of air inside this. So that is called as dead space volume. Now do not confuse this with all these 4 lung volumes. Okay. Now let's talk about capacities. Now with the help of these 4 lung volumes, you can calculate capacities. So first capacity is IC, inspiratory capacity. It's very easy. If you add tidal volume and inspiratory reserve volume, you are gonna get inspiratory capacity. If you wanna get expiratory capacity, you have to add tidal volume and expiratory reserve volume. If you want to get functional residual volume FRC, you have to incorporate, you have to add expiratory reserve volume with residual volume. And most importantly, vital capacity. So if you remember this term, Virat wears a tie. So Virat for vital capacity, tie, tidal volume, inspiratory reserve volume, expiratory reserve volume. If you add these 3, then you are gonna get your vital capacity. And obviously the last one, that is TLC, total lung volume, you have to add all the 4 volumes you have to add all the 4 volumes, TV plus IRV plus ERV plus RV, or just you can add vital capacity plus residual volume, then you are getting your total lung capacity. Yes, oxygen hemoglobin dissociation curve is your weak point. Obviously we are going to discuss each and every thing from this chapter. Okay, so this is a diagrammatic representation that if you consider your lung as an empty balloon kind of structure, the minimal air always remain inside your lung, that is called your residual volume. 500 ml you can exhale or you can inhale, that's your tidal volume. 500 ml you can exhale or you can inhale, that is your tidal volume. And expiratory reserve volume is the air that can be exhaled forcefully. Expiratory reserve volume is the air that can be exhaled forcefully over a normal exhalation, and the volume of air can be inhaled forcefully over a normal inhalation, that's your simply inspiratory reserve volume. So, whenever we are talking about inspiratory reserve volume or expiratory reserve volume, we have to take consider of that forcible part. Inspiratory reserve volume means forcible inhalation, expiratory reserve volume means forcible exhalation. Okay. Now, I will be talking about exchange of oxygen and carbon dioxide and various factors that affecting the mode of the diffusion from from alveoli to blood or blood to alveoli. Let us talk about that. Okay. So, the factors which are affecting the exchange of gases, they are obviously pressure gradient, means obviously the gases gonna move or will show their movement from high pressure zone to low pressure zone, that is called pressure gradient. Secondly, the solubility of gases, if the gas is more soluble, then there will be tendency to the faster diffusion, right. And we know that carbon dioxide is 20 to 25 times means carbon dioxide is having 20 to 25 times more solubility than oxygen. So, if you take a unit surface area and unit time, and if you want to diffuse, if you are punching carbon dioxide and oxygen inside the water, so the more number of carbon dioxide molecules will be diffusing or getting dissolved into the solution or into the water. Third factor affecting exchange of gases from one place to another place, that is thickness of membrane. So, thickness, if it is less thick, membrane is less thick or thin, then there will be more chances of exchange of gases. If the thickness is more, then there will be less chances of exchange. So, exchange of gases is inversely proportional to the thickness of membrane. Exchange of gases is inversely proportional to the thickness of membrane. Less thin, less thick, more exchange, more thick, less exchange. Okay. And you know that thickness of the diffusion membrane should be less than 1 millimeter, it is near about 0.2 millimeter. Now, let us elaborate the thickness of membrane first. So, if we take consider our alveolar surface, if this is the alveoli, then the alveoli is mostly made up of some simple squamous cells called as pneumocyte 1. The alveolar epithelium is made up of simple squamous epithelium, right. And in between them you may find some cuboidal or columnar cells as well, or some oval shaped cells as well, but mostly the cells are of What shape? Cells are of simple squamous epithelium shape, and the cells are called as pneumocytes. So the cells of alveoli, we call them pneumocyte cell. Okay. Now let's come towards the surrounding blood vessel of alveoli. Alveoli are rich in blood supply. They are surrounded by blood capillaries. So the lining, cellular lining inside the blood capillary, they are also simple squamous epithelium. They are also simple squamous epithelium. Obviously, you know that squamous epithelium means they are very much thin and flat, and in between them, in between the blood capillary and alveoli, we are getting basement substance. We are getting basement substance which is making basement membrane. So there are three layers. If air is going to come from alveoli to blood, then it has to cross three membrane. First, alveolar epithelium. Second, basement substance. What is basement substance? Non-cellular jelly-like space between alveoli and blood vessel. And the third layer is epithelium or endothelium, means lining of blood capillaries. Endothelium of blood capillary. So you have to cross these three layers if you want exchange of gases, and NCERT is saying that if you consider all these three layers, the thickness should be less than 1 millimeter. So that was all about thickness of membrane. Now let's focus on pressure gradient of gases. So this chart or this table is already given in NCERT. We have to take care of partial pressure of oxygen and partial pressure of carbon dioxide. Now, what do you mean by partial pressure? Partial, yes, pneumotaxis center I am coming when we will be talking about regulation of respiration, right. So what is partial pressure of gases? See, air is mixture of many gases. Air has 78% nitrogen, 21% oxygen, 0.03% of carbon dioxide. Yes. So that is called atmospheric pressure. Now, what percentage of oxygen is present into air? 21% of oxygen is present into air. What percentage of carbon dioxide is present into air? 0.03% of carbon dioxide present into this air. So if air pressure is 760, you just have to calculate only 21% of that, and you will be getting partial pressure of oxygen. Means the part pressure, which is exerted by oxygen in a unit surface area. The part pressure of carbon dioxide, that is only 0.03% of 760 mm mercury pressure, then you will be getting 0.3. So you might be thinking, so how to calculate the partial pressure of oxygen in air and how to calculate the partial pressure of oxygen, partial pressure of carbon dioxide in air, right. So if you are unable to memorize it, just remember what is the total pressure of air. What is air pressure? 760 millimeter mercury pressure, right. Now what is the percentage of oxygen in air? Oxygen is 21% and carbon dioxide is 0.03%. So what you have to do, you have to perform simple mathematical calculation. You just try to calculate 21% of 760, the answer you will be getting nearby 159. You want to calculate 0.03% of 760, the answer you will be getting somehow 0.3 mm mercury pressure. So that's how we have calculated the partial pressure of oxygen and carbon dioxide in ATM or atmospheric pressure. Okay. Now from atmosphere, oxygen is going towards alveoli. Why? Because the partial pressure present inside the alveoli, the oxygen is 104 mmHg, and you know that a gas will always be traveling from high pressure to low pressure. So oxygen is traveling from atmosphere to alveoli. Now towards your alveoli, deoxygenated blood is coming, and the partial pressure of oxygen in deoxygenated blood is only 40 mmHg. Now alveoli will compare, I have partial pressure of oxygen 104 and the deoxygenated blood which is coming to me, that is having partial pressure of oxygen only 40. So I have to travel from alveoli towards the blood, and oxygen will enter into deoxygenated blood, and that will make the deoxygenated blood into oxygenated one. And that is the reason oxygenated blood is having 95 mm oxygen, 95 mm Hg partial pressure of oxygen. Okay, now this oxygenated blood is going towards your tissue, and your tissue is having very low partial pressure of oxygen, only 40 mmHg. So what this oxygenated blood will do? Oxygenated blood will see, I have 95 partial pressure and the tissue is having only 40 partial pressure. So according to the rule, I have to move from high pressure to low pressure. So oxygen will travel from oxygenated blood to tissue to perform the cellular respiration or oxidation. Now let's check for carbon dioxide. Inside your cells, respiration is going on, carbon dioxide is getting produced. So that's why tissue is having high partial pressure of carbon dioxide, that is 45 mmHg. Now while this oxygenated blood will come towards the tissue to deliver oxygen, the tissue will see, the carbon dioxide will see that I have more pressure and the blood is having only 40 partial pressure of carbon dioxide. So carbon dioxide will move from tissue to oxygenated blood, and that extra carbon dioxide will increase the partial pressure of your blood, and that will make your blood deoxygenated. Now your blood is having 45 mmHg mercury pressure of carbon dioxide. Now this deoxygenated blood will go towards your alveoli, and it will observe the partial pressure of carbon dioxide is only 40 inside alveoli. Now again, following the rule, carbon dioxide will move from 45 to 40, means high pressure, to low pressure. Now you have 40 mmHg mercury pressure of carbon dioxide inside your alveoli, and that will check that in atmosphere the partial pressure of carbon dioxide is very low, 0.3. So again, from alveolar air the carbon dioxide will move outside. That's why we say that during the time of inhalation we intake oxygen, and during the time of exhalation we exhale carbon dioxide. That is the reason, that is the technical reason if you observe this particular chart. Now one trick I am saying, if you are forgetting what should be the value, atmospheric air, alveoli, deoxygenated blood, oxygenated blood, and tissue. Now we all know that atmospheric of PO2 is 159 and atmospheric pressure of PCO2 is 0.3. Okay. Now from alveoli to tissue, everything is inside our body. Now remember, inside our body, if it's weaker than the other, if oxygen is weaker or carbon dioxide is weaker, then the partial pressure will always be 40. Now let me discuss that inside alveoli, who is stronger and who is weaker. Inside alveoli, oxygen is stronger, carbon dioxide is weaker. So the partial pressure inside the alveoli for carbon dioxide will be 40. Inside deoxygenated blood, who is stronger? Deoxygenated, carbon dioxide is stronger. So who is weaker? Oxygen is weaker. So inside deoxygenated blood, the partial pressure will be 40. Inside oxygenated blood, oxygen is stronger, carbon dioxide is weaker. So inside oxygenated blood, the partial pressure of carbon dioxide will be 40. Inside tissue, who is stronger, who is weaker? Tissue is producing ample amount of carbon dioxide. So carbon dioxide is stronger and oxygen is weaker. Inside tissue, the partial pressure of oxygen will be 40. Okay. Now, inside our body, if carbon dioxide is strong than oxygen, then the partial pressure of carbon dioxide is gonna be 45. So in tissue, who is strong? Carbon dioxide is strong. So the partial pressure of carbon dioxide will be 45. Inside deoxygenated blood, who is strong? Carbon dioxide is strong. So the partial pressure of carbon dioxide will be 45, and for alveoli and oxygenated blood you have to remember into separate manner. For alveoli, partial pressure of oxygen is 104, and for oxygenated blood the partial pressure of oxygen is 95 mmHg. So that's how you can recall this table given in our NCERT, and we are done with this slide. Now we will be moving forward. Whatever we were discussing, from where the air is traveling from which place, this diagram is justification of that only. So I won't be detailifying much about that. So directly we will be talking about transportation of gases into our body. Okay. So we have two gases to transport. One is oxygen and another is carbon dioxide. Now when you will load your blood with oxygen, when your blood is oxygenated, when oxygen is entering into your blood, then how your blood will be carrying oxygen? So it's very easy to remember. 97% of your oxygen will go with RBC, and you know that RBC is containing the volume of RBC is occupied by hemoglobin. 36% of volume of RBC, inside your RBC, the 36% area, 36% volume will be only hemoglobin. So that will make oxyhemoglobin, a temporary compound. So oxygen will go inside RBC and will form oxyhemoglobin. There will be temporary coordination bond. There will be formation of temporary coordination bond in between the iron of hemoglobin and the oxygen molecules, and that compound is called as oxyhemoglobin. So 97% oxygen is transported as oxyhemoglobin, and only 3%, the remaining 3% oxygen will be transported as dissolved condition directly into your plasma, because plasma of your blood is nothing but 90% of water, right? So inside that water, 3% will be easily dissolved. Okay. Now let's come to carbon dioxide. Now again, 70% of your carbon dioxide will be traveling into plasma, but not in dissolved format as sodium hydrogen carbonate, that is with bicarbonate. So 70% of carbon dioxide, when carbon dioxide is getting entry into your blood, that carbon dioxide will become bicarbonate ion, and that bicarbonate ion, HCO3 minus ion, that HCO3 minus ion will react with sodium present into your plasma. Your blood has lot of salt molecule, right? NaCl. So that Na plus will react with HCO3 and that will form sodium hydrogen carbonate, and that's written majority of carbon dioxide is transported as bicarbonate. Now only 20 to 25% carbon dioxide will get inside RBC and remain inside RBC, and by holding the tail of hemoglobin molecule, and they will form carbaminohemoglobin. 20 to 25% will converted into carbaminohemoglobin, or will be traveling with hemoglobin molecule, and remaining 7%, as because carbon dioxide is having 25 times higher solubility than oxygen. So here 7% carbon dioxide is getting dissolved into plasma. So overall, this is the summary of gaseous exchange, or sorry, transportation of gases. Now first of all we will be talking about transportation of oxygen molecule inside our body. Okay, so behind the transportation of oxygen, who is responsible? It's hemoglobin. It's hemoglobin. Now very interesting fact I am telling, like if I give you two option, first option you have to study whole night today, and second option you have to play game. Okay, any favorite game of you whole night today. So what you will be choosing? Obviously you will be choosing playing game. Okay, in spite of having your exam and knocking at the door, right? So similarly you will be choosing the bad thing. So suppose you are the hemoglobin molecule and you have to choose between oxygen, carbon dioxide and carbon monoxide. See what you are gonna choose? You are gonna choose the carbon monoxide, the harmful molecule for you. So the affinity of hemoglobin is highest for carbon monoxide, then it will be for oxygen, and then it will be for carbon dioxide, right? So when hemoglobin binds with carbon monoxide, the hemoglobin is spoiled. We call the molecule, call the component as carboxyhemoglobin, right? So basically most of the time the hemoglobin is permanently damaged if your hemoglobin is binding with carbon monoxide, and that may cause death of you, right? If hemoglobin is binding with carbon dioxide, then it will form carbaminohemoglobin. It's a temporary component, it's not a permanent component. And next, if your hemoglobin is binding with oxygen, that will form oxyhemoglobin. And remember, we know that if one component or if one molecule is taking oxygen with it, generally that is, we say that phenomena as oxidation. But here in this case of oxyhemoglobin formation, hemoglobin plus oxygen, that's not gonna be oxidation reaction. We call this as oxygenation. We call this as oxygenation. Why it's so? If there is formation of a permanent compound, then it's said that is oxidized by oxygen. But when oxygen is binding with hemoglobin, that's a temporary bonding, it's kind of coordination bond, right? And that's why instead of saying oxidation, we are saying this as oxygenation. Okay, and you need to remember this simple calculation, that your 100 ml of blood contains 12 to 16 gram of hemoglobin. Generally males, inside male blood, the amount of hemoglobin is little bit higher in compared to female. Okay, in male 100 ml blood is containing 16 gram of hemoglobin, in female 100 ml blood is containing 10 to 12 gram of hemoglobin. Now those who are watching this, can you please comment and give me the answer, why it's so that hemoglobin content, RBC content, or the blood content, the volume of blood, the volume of RBC or hemoglobin is more in male body in compared to female body? Can you please write down into the comment section? You might have seen that an average male is containing near about 5.5 liter of blood, but that female, made for hard working. Okay, so if a female is working more in compared to a male, then obviously then she will be getting this like amount of hemoglobin similar to male. Will it be like that, or is there any other reason? Can anybody answer me? Why the hemoglobin component or RBC or blood is found in higher amount in male body in compared to female? What is the reason? It's not hard working. Okay, let me answer this. It's because uh, see, you have to talk about the RBC producing hormones. It's erythropoietin hormone, it's thyroid hormone, it's cortisol hormone. So erythropoietin, cortisol and thyroid hormones are produced in both the cases, in both the sexes, in males as well as in females. But if you talk about testosterone or the androgens hormones, the testosterone is produced in higher amount in case of males, but in negligible amount in case of females. So that is giving the added advantage to a male in compared to a female. So testosterone is also one of the hormone which is responsible for blood formation or RBC production. So that's why overall if you check, female is having 3 hormones for RBC production: cortisol, thyroid and erythropoietin, but male is having 4 hormone: testosterone, cortisol, thyroid and erythropoietin. That is the reason. Okay. So overall 100 ml of blood is containing 12 to 16 gram of hemoglobin, and one hemoglobin molecule can carry 4 oxygen. One hemoglobin molecule can carry 4 oxygen. Why it's so? Because hemoglobin is made up of 2 alpha chain and 2 beta chain with a porphyrin ring that is having 4 Fe2 plus, right? Hemoglobin is having 4 Fe2 plus, and each Fe2 plus can bind with one molecule of oxygen. So 4 Fe2 plus inside one hemoglobin can bind with maximum 4 molecule of oxygen. So if 4 molecule of oxygen is binding with one hemoglobin molecule, then we will be saying the hemoglobin is 100% saturated. If 3 molecule of oxygen is binding with a hemoglobin molecule, then we will be saying that hemoglobin is 75% saturated. If 2 oxygen molecules are binding with one hemoglobin, then we will be saying that hemoglobin is 50% saturated. So one hemoglobin molecule at the highest level, maximum level, can carry 4 molecules of oxygen, and if we calculate, so 1 gram of hemoglobin will be carrying 1.34 milliliter of oxygen, and as we know that 100 ml of blood contain 12 to 16 gram of hemoglobin. If you multiply 15 gram of hemoglobin into 1.34, somehow you will be getting 20. So that's why it's written here 100 ml of oxygenated blood will be carrying 20 ml of oxygen. So this chart or this calculation simply you have to remember, right? Okay, now we will be talking about oxygen hemoglobin dissociation curves and Bohr's effect. Okay, a few tricky topic, or you can say some difficult topics, the most difficult topic of this chapter. Okay, this oxygen hemoglobin dissociation curve. So you need to remember this, that hemoglobin and oxygen, they are binding with each other to form oxyhemoglobin. Hemoglobin and oxygen is a temporary binding coordination bond, and there will be formation of oxyhemoglobin. So if hemoglobin is carrying oxygen, so that will be called as association or binding, and if hemoglobin is losing, kicking out oxygen from it, then we will be calling that phenomena as dissociation. So when this compound is formed, then that is called association or binding. When opposite thing is happening, oxyhemoglobin is broken into oxygen and hemoglobin, then that phenomena is called as a dissociation. So what are the important factors will be influencing either hemoglobin will be binding with oxygen or hemoglobin will do a breakup with oxygen? Either it's patch up or break up. What are the factors, what are the components or the situation inside our body going to influence this? First, partial pressure of oxygen can influence, partial pressure of carbon dioxide can influence this, temperature can influence this, H plus ion concentration can influence this, okay, acidity can influence this, When you are talking acidity, obviously pH can influence this, and one component called DPG, diphosphoglycerate, one acid can influence this. So these are the components which can influence either hemoglobin will be binding with oxygen or hemoglobin will do breakup with oxygen. So if partial pressure of oxygen is okay, so I am making one signal. If love signal is there, that means hemoglobin and oxygen binding. If love signal, if broken heart is there, that means dissociation of hemoglobin and oxygen, right? So if you observe high partial pressure of oxygen, that will create what? That will create oxyhemoglobin or binding of hemoglobin and oxygen. If low partial pressure is there, then that will cause breakup between oxygen and hemoglobin. Now let's talk about carbon dioxide. If high PCO2 is there, then that will cause dissociation. If low PCO2 is there, that will cause association. If high temperature is there, that will cause heartbreak, that means breakup between hemoglobin and oxygen, dissociation. If low temperature is there, then that will cause binding of hemoglobin and oxygen. Okay. If high percentage or high concentration of H plus ion is there, that will increase acidity, and acidity will do breakup between hemoglobin and oxygen or dissociation. If there is low H plus concentration, that will promote binding of hemoglobin and oxygen. If acidity is high, means more H plus ion, that will cause dissociation, means breakup between hemoglobin and oxygen. If acidity is low, low acidity means towards alkalinity, towards basicity, that will promote binding of hemoglobin to oxygen. If high pH, if pH crosses 7, if pH crosses 7.4, 7.6 and 8, then there will be more chance of formation of oxygen and hemoglobin. So patch up between hemoglobin and oxygen will be there. If low pH is falling below 7.4, then there will be breakup between hemoglobin and oxygen, that means dissociation will be there. And lastly, DPG, a product of your respiration, if DPG is produced in a higher amount in your body, that is obviously not good for oxyhemoglobin, that will be promoting dissociation. If there is low concentration of DPG into your blood, into your body or into your tissue specifically, that will promote association of hemoglobin and oxygen. So again, heart means patch up between hemoglobin and oxygen, binding of hemoglobin and oxygen, affinity towards, good affinity of hemoglobin towards oxygen. Broken heart or crossed heart, that means dissociation of hemoglobin and oxygen, and these are the factors which can influence either hemoglobin will be making bond with oxygen or hemoglobin will be breaking bond with oxygen. Okay. DPG means diphosphoglyceric acid. Sometimes it's written as BPG, bisphosphoglyceric acid. It's a product of your respiration, right? So if there is more respiration, more carbon dioxide, more acid and more DPG. Okay, and that is the reason we are saying that if we take a graph, x axis tells us about partial pressure of oxygen and y axis tells us about hemoglobin and oxygen saturation percentage, means how much hemoglobin is carrying what percentage of oxygen into our body. Okay. Now you need to know that maximum 97%, maximum 97% of oxygen can be transported by hemoglobin. So the graph, if you make the graph like this, gradually you are increasing the partial pressure, the graph will create a slope and steep, and gradually towards 97% the graph will become linear. The graph never cross the 97% threshold limit or saturation limit. Very first question can be asked, the type of this graph. Type of this graph is sigmoid. What do you mean by sigmoid? Sigmoid means S-shaped. Sigmoid means is a kind of S-shaped curve. So the oxygen hemoglobin dissociation curve is S-shaped. Now why it's S-shaped? See, this axis you are increasing the partial pressure of oxygen gradually. X-axis is telling us you have to increase the partial pressure. So initially when you were increasing the partial pressure, the graph was too much slopey, means the saturation or the binding between oxygen and hemoglobin was very slow, that's why it's forming a slope. But when it crossed a particular limit, particular partial pressure of oxygen, gradually the hemoglobin is become very fast to bind with oxygen. So that's why it's creating a steep region. So the steep region tells us very fast binding of hemoglobin and oxygen. The slopey region tells us slow binding of hemoglobin and oxygen, and when it's going towards, approaching towards 97%, again it's becoming slopey. So first slope, then steep, then again slope. That is the sigmoid nature of the curve. That means you can justify by saying, initially if you increase the partial pressure of oxygen, hemoglobin is showing low affinity towards oxygen. It's very slowly binding oxygen with it. Initially, if you are increasing the partial pressure of oxygen, but when the oxygen partial pressure has crossed a particular limit, suddenly, very happily, more hemoglobin is very fastly started binding oxygen molecules with it, and that's why the graph is rising by forming a steep region. And once it's approaching towards the saturation, is simply when you start eating something, you will be eating your favorite food. Suppose my favorite food is biryani. So when I have started eating, I was quite fast while I was eating biryani. But as soon as I felt my stomach is getting gradually full, I will stop the speed of eating. Similar thing is observed here. When the hemoglobin is gradually binding the oxygen, but it's approaching towards the saturation level, the binding sites are becoming limited. Again it's forming a slopey nature, a slopey part of the curve, and by when it's reaching the saturation, means if my stomach is full, if hemoglobin's binding site is full, upon coming into the 97%, no more oxygen cannot be binded with hemoglobin. So near to the 97%, it's forming a linear line. So that is the justification of this sigmoid nature of the curve. And this phenomena, if you increase partial pressure, affinity will also increase. That is called cooperative binding of hemoglobin and oxygen. Cooperative binding of hemoglobin and oxygen. Okay, let me give you, let me simplify it in another manner. Suppose I have a deal with you that I will be paying you money for studying. Okay, so I have started paying you. So I have given you 100 rupees. You are not quite keen, then you are studying per day for 1 hour only. Now I have given you 200 rupees. Then you are studying for 1 hour and 10 minutes. Then I have given you 500 rupees. You said, what I will do with this 500 rupees? Okay, I will study for 2 hours per day. But the next day, when I have started giving you 2000 rupees per day, then you are studying for 5 hours a day, right? So with money, while I have increasing giving you money, you have increased your study hour. The next day I have given you 5000 rupees. Now this day you are studying for 10 hours. Now that is your highest limit. The next day I have given you 10,000 rupees for studying, but you said that's my maximum limit. Beyond 10 hour I cannot sit and read and write. So next day also you will be studying for 10 hours only. So that's the same phenomena can we observe, the binding affinity in between hemoglobin and oxygen. So why it's called as dissociation curve? Why it's called as dissociation curve? It's called as dissociation curve because, see, your alveoli, near to your alveoli, your blood will be loading all the oxygen, and gradually by following the circulation mode, your body is gonna circulate and unload those oxygen to your different tissue. Now your different tissue will be seeking for different percentage of oxygen. If you are hungry for 5 hours, you will, suppose if I give you roti to eat, you will eat 5 rotis. If you are hungry for 1 day, then you may eat 10 rotis, and if you are hungry for 2 days, you may eat 15 rotis. So similarly, different tissue of your body is performing different work. So few are seeking for more oxygen and few are seeking for less oxygen. So how your body is gonna decide that? Your body will be deciding by checking the partial pressure of oxygen into that particular tissue, right? If your body observe that one tissue is having 40 partial pressure of oxygen, 40 mmHg of partial pressure of oxygen, then according by following the graph, your body will be dissociating 75% of oxygen and your body will be keeping 25% of oxygen with blood. If your body finds that tissue with 60 partial pressure of oxygen, then how much oxygen your body will be keeping and how much oxygen your body will be dissociating? Then in that case your body will be keeping near about 90% of oxygen and will be dissociating only 10% of oxygen. Okay, if your body observe a tissue is having 40 partial pressure, then your body will be dissociating 25% of oxygen and will be keeping 75% of oxygen. If your body observe a tissue with 20 oxygen partial pressure, then your body will be dissociating near about 25%. If your body will, then your body will be dissociating near about 75% of oxygen and will be keeping only 25% of oxygen. So these graphs will be helping your body to decide, depending upon the necessity, when to give more oxygen and when to give less oxygen to your different tissue. Like when you are solving a very tough mathematical solution, then which organ is working effectively? Then your brain is working effectively, then your brain will be needing more volume of oxygen, right? But while you are doing exercise, while you are running, then your muscles will be needing more oxygen than your brain. So that is the importance of this oxygen hemoglobin dissociation curve. Okay. Now the next question can be asked, sir, when to do right shift of the curve and when to do left shift of the curve? Just remember that right shift means something bad is happening inside your body, and left shift means your condition, body condition is very good. Suppose you faced a massive accident, or you are suffering from fever, or let me come into that particular topic. Right now I am discussing when your body, this curve will be doing left shift and when your body will be right shift. When there is a bad condition, right? If you are suffering from fever, if you have faced a massive accident, right? Or if you are sitting in a room where the room is totally crowded, you are suffocating, then in those conditions the curve will be doing right shift. But suppose you are standing into a near to a ocean or sea, wind is coming, ample amount of oxygen is there, no one is there, a very silence, calm and a cool beach is there, sea beach. Okay, you have gone for a vacation. So in that particular condition you are feeling healthy, you are feeling charmed, so the curve will be shifting towards the left. So left shift of the curve means good condition, right shift of the curve means bad condition. So we have already checked about the factors which can affect. So right shift means broken heart, left shift means full heart, the right shift means hemoglobin and oxygen dissociation, low affinity of hemoglobin and oxygen. Left shift means hemoglobin is happily binding with oxygen, high affinity of hemoglobin and oxygen, right? So that's the reason high partial pressure of oxygen, low partial pressure of carbon dioxide, low concentration of H plus ion, low acidity, high pH, low temperature, low amount of BPG or DPG will promote left shift of the hemoglobin oxygen dissociation curve, but the opposite condition, low partial pressure of oxygen and high partial pressure of carbon dioxide, high concentration of H plus ion, high temperature, low pH, high acidity can give you a right shift of oxygen hemoglobin dissociation curve. Gary is asking who motivates you. There are many people around me motivating me. Firstly, obviously, my father, who used to motivate me a lot in my life, but my father is no more. Then my wife is also very helpful. My wife is motivating me every day for my daily life, to pursue my journey, to pursue my dream. And obviously you students, okay, students also motivates me every day. See, a good students will be making a good teacher, and a good teacher will be making a good student. So it's a both and journey, it's a vice versa journey. So a teacher motivates his or her students, and students also are similarly responsible for motivating their teachers, okay, their mentors. So that's all. Now Gary, yeah, Gary was saying, sir, repeat this particular graph one more time. Okay, so again I am repeating it. Whenever you are studying this oxygen hemoglobin dissociation curve, how to study this, how it's affecting our body condition. So basically this curve is telling your body circulation, your circulatory system, that based upon the need you have to supply oxygen into different tissue. I am giving you one more example. You are sitting with some food and you want to give the food to some needy person. Okay. Now already I am coming to you by eating 2 plates of chicken biryani. So will you be giving me that food? No. But along with me one beggar is coming to you who has not eaten for 2 days. So it's your duty. What will be your duty? Your duty will be to give that food to that beggar, to the needy person. Similarly, your near to alveoli, your blood is becoming oxygenated. Near to alveoli, your blood is becoming oxygenated. near to your lung alveoli, your blood is becoming oxygenated. That means your blood is having 100% of oxygen into it. Now you have two different tissue into your body. You have three different tissue into your body. For first tissue, tissue A, the tissue is working very hard and the partial pressure of oxygen is very low into that tissue. The partial pressure of oxygen is only 20. You have another tissue into your body which is working moderately. Moderate partial pressure of oxygen is there and the partial pressure of oxygen is 40 mmHg for tissue B, and for tissue C it's chilling inside your body, it's really not doing any work for you for the time being. So suppose for that particular tissue the partial pressure of oxygen for that particular tissue is 60. Now you have to decide, your circulatory system has to decide where to supply what percentage of oxygen. So how your body gonna decide? Your body will be following this chart. See, for one tissue the partial pressure is 20. So for one tissue the partial pressure is 20. So here what your body will do? Your body will keep 25% of oxygen and donate 75%. So for this particular tissue your body will donate 75% of oxygen by keeping only 25%. Now your blood has gone to another tissue and has seen the tissue is having partial pressure of oxygen as 40. Your body will calculate this curve. So at partial pressure of 40 your body can retain 75% of oxygen and can donate 25% of oxygen. So here your body will be donating 25% of oxygen but retaining 75% of oxygen. The tissue C, the partial pressure of oxygen is 60. So at partial pressure of oxygen where 60, your body will calculate that your body can retain 92% oxygen and donate 8% oxygen. So here what your body will do, your body will retain 92% oxygen and will only donate 8% of oxygen to this particular tissue, and that's how justice is served. That's how justice is served. If your tissue is needing for the oxygen, if your tissue is going out of oxygen, like you can see many of the families going out of gas cylinder light, right? So if your tissue is going out of oxygen, the oxygen partial pressure is very low, the tissue is crying by saying hypoxia, then your body gonna deliver 75% oxygen to that tissue. If your tissue is sitting with partial pressure of oxygen 40, in normal condition, normal physiological condition, this is called the partial pressure of oxygen in your tissue in normal physiological condition, then your blood will be donating 25% of oxygen to that tissue by keeping 75% as a reserve. Okay, now the next topic is transportation of carbon dioxide. See, transportation of carbon dioxide is a very easy phenomena to remember. So basically this red oval shaped structure, this our RBC. So what's happening here, from your tissue, from our tissue, the carbon dioxide is entering into our blood. From our tissue the carbon dioxide is entering into our blood. Okay, upon entrance 7% of carbon dioxide is directly getting dissolved. Upon entering into the blood, 7% of carbon dioxide is directly getting dissolved into plasma of your blood. Okay, next 70% of carbon dioxide is entering into your RBC. 70% of those carbon dioxide after entering into your RBC, red blood corpuscle, it's reacting with water. So CO2 plus H2. Now who is influencing this reaction, who is catalyzing the reaction? Ca, carbonic anhydrase. Carbonic anhydrase is special catalytic enzyme present more into your RBC. But if you get a statement like this, in our body carbonic anhydrase enzyme is present only inside RBC, that's a wrong statement. RBC is rich in carbonic anhydrase enzyme, but also your plasma is having very less amount of this carbonic anhydrase enzyme. So upon entering by the influence of carbonic anhydrase, CO2 and H2O will react together to form carbonic acid H2CO3. Again this carbonic anhydrase, the catalyst will come and break it down H2CO3 into a proton, means H plus ion, and HCO3 minus. See, now what will happen? This proton H plus ion is a monkey kind of ion. It will be jumping here and there and try to disturb RBC. Now what your RBC will do? RBC will scold this H plus ion and told, don't behave like monkey, hey monkey fellow. Now I need to stop you from doing mischievous activity. Now this H plus will be trapped by hemoglobin present inside RBC, and when this H plus is trapped by your hemoglobin, that will make your hemoglobin acidic and that will form hemoglobinic acid. And the remaining HCO3 minus, that will be kicked out, from your RBC into your plasma with the help of a special protein present into the cell membrane of your RBC. Okay, now once HCO3 minus is kicked out from RBC to plasma, that HCO3 minus will react with Na plus ion, which is present already into your plasma. Na plus, see, into your plasma you have Na plus ion and Cl minus ion, right? Now by observing HCO3 minus, Na plus will break up with Cl minus and will bind with HCO3 and forming sodium hydrogen carbonate, NaHCO3, and that's how 70% carbon dioxide is stabilized into your plasma. Your blood will be carrying 70% of carbon dioxide as NaHCO3, and once Na plus has done breakup with chloride ion Cl minus, the RBC will happily call it back into inside because it has kicked out another negative ion. So this exchange of two negative ions, kicking out HCO3 minus outside and welcoming chloride ion inside RBC, is called as chloride shift, and it was observed by a scientist called Hamburger. That's why we call this phenomena as Hamburger's effect. Okay, now upon getting inside, Cl minus will react with potassium ion, K plus Cl minus, that will form KCl, temporary component inside the RBC. Okay, now here what's happened with the hemoglobin molecule? By catching this H plus ion, hemoglobin became acidic in nature, hemoglobinic acid. Now this acidic hemoglobin is not capable of retaining oxygen with it. So this acidic hemoglobin, what it will do, it will release the oxygen from it, and the oxygen will enter into the tissue to perform cellular respiration. Now once oxygen is kicked out from hemoglobin, carbon dioxide will gradually build affinity with that acidic hemoglobin or hemoglobinic acid, and carbon dioxide will go and bind with the tail of the globin chain of hemoglobin, and that's why it's occupying one of the amino group of the globin chain. That's why it's called as carbaminohemoglobin. See, hemoglobin is having globin chain, and globin chain is what? Protein, and protein is made up of amino acid. Amino acid is having amino chain, so the carbon dioxide is going directly binding with the amino group of the one of the amino acid of the globin chain of hemoglobin. That's why we call carbon dioxide plus hemoglobin as carbaminohemoglobin, and as carbaminohemoglobin, 25% carbon dioxide is settled inside RBC. So that is how your deoxygenated blood is carrying carbon dioxide into it. Now we are moving forward to the next part of this chapter, that is regulation of our respiration. It's very super easy topic, regulation of respiration. See, the main center of regulation of respiration, that is situated into our medulla oblongata region of our hindbrain. Medulla oblongata, the last part of your brain, right? And it has a special center called respiratory rhythm center, shortcut I say it RRC. So this respiratory rhythm center is mainly responsible for maintaining your normal inspiration and expiration. Okay, that's one thing, that's the most important thing. Rest. Okay, we have few other parts present into our brain that can give signal to your respiratory rhythm center. Either we have to increase the rate of breathing or we have to decrease the rate of breathing. See, breathing is directly regulated by respiratory rhythm center, but we have few other centers present into our brain which can give order or give signal to that respiratory rhythm center that when we need to increase the rate of breathing and when we need to decrease the rate of breathing, and those centers are present into our pons region of hindbrain, right? And we have two such center. One center is called as pneumotaxic center, and the pneumotaxic center is responsible for increasing the rate of breathing. Pneumotaxis means increase the rate of breathing by decreasing the time duration of inhalation. See, if you are taking deeper inhalation, deeper exhalation, that will take more time. But if you are doing faster inhalation and faster exhalation, so that will decrease the depth of your breathing but increase the rate of your breathing. So what pneumotaxis center does for you? Pneumotaxis center increase your rate of breathing. So what is the normal breathing rate? 12 to 16 times per minute. So upon the influence of pneumotaxic center, you can breathe at a rate of 20 or 25 inhalation and exhalation per minute. But the opposite phenomena is done by apneustic center. Apnea means absence of breathing. Apneustic center is responsible for decreasing the rate of breathing and increasing the depth. Means if you are under control of apneustic center of your pons, then you are going to take long breath. Slow breathing will be there. If you are under control of pneumotaxic center, fast breathing will be there. So that's why we call this as moderator of breathing activities, pneumotaxic center and apneustic center. Okay, and along with this, we have some chemosensitive areas. We have some chemosensitive areas associated with our body, different parts of our body. They can give signal to the respiratory rhythm center. Either we have to increase or decrease the rate of breathing. So those chemosensitive area, they are sensitive only for carbon dioxide and hydrogen ion. They cannot sense oxygen really. So the chemical sensors present into near your respiratory rhythm center, inside your aortic bodies, means inside your aorta, and inside your carotid bodies present inside your carotid artery. So those chemosensitive areas are present where? Few bodies are present into your arteries, like aortic bodies inside your aorta, the main artery of your body, and carotid bodies, the artery which is taking oxygenated blood to your brain. And one such area is present near to you, just adjacent to your respiratory rhythm center. while blood is circulating into your blood vessels, they are just checking the presence of carbon dioxide and H plus ion and giving the signal. If more carbon dioxide and more H plus ion is there, that means your blood is acidic, you need to increase the rate of breathing, that means you need to inhale more and more oxygen. So that's how your body is performing or regulating your breathing. And remember this line, role of oxygen is insignificant in regulation of respiration. Role of oxygen is insignificant in the regulation of respiration. Now in higher class you will study one more thing, that is called hearing brewer reflex. That means if you keep inhaling more and more air, if your lungs and respiratory tract is inflated, if your respiratory tract is inflated and stretching, then automatically your body will give you signal to release the air or to perform exhalation. That's why there is a limitation that you can hold your breath, and that reflex is called as hearing brewer reflex. But for NCERT, if you stick to the NCERT, you don't have to study that. You need to remember about this respiratory rhythm center, this chemosensitive area. How many chemosensitive area we need to talk about? One area near to respiratory rhythm center, aortic bodies present inside your aorta, carotid bodies present inside your carotid arteries. And do not forget about, there is no significant role of oxygen is there. So for chemosensitive area is the carbon dioxide and H plus ion who can exert sensation to your chemosensors. Now let's talk about some disorders. So first disorder or first respiratory disorder, that is asthma. See, what is asthma? Asthma is kind of allergy. Asthma is kind of allergic reaction, and it is defined by difficulty in breathing with wheezing sound. Basically what happens, there is inflammation in your bronchi and bronchioles, inflammation of your bronchi and bronchioles. This is the area inside your normal bronchi, and this is the area inside your infected bronchi. If there is allergic reaction happening inside your bronchi, then the muscles will be starting releasing fluid materials, and the bronchi will start getting choked. The area, the lumen of your bronchi will become narrow, and that will cause difficulties in taking air, and that's why you will be making that wheezing sound. This kind of sound will be made by you, by your respiratory tract, and that's called wheezing sound. So remember, difficulty in breathing, wheezing sound, and it's due to the inflammation in bronchi due to the excess secretion of histamine. Remember. Next disorder is emphysema. Emphysema is also sometimes called as COPD, chronic obstructive pulmonary disorder, chronic obstructive pulmonary disorder, emphysema. If you are long-term or chain smoker of cigarette or similar kind of thing, then what will happen? The particles of the cigarette smoke, they are gonna affect your alveolar wall, and when your alveolar cells, remember the pneumocyte cells, when those pneumocyte cells are damaged of your alveoli, the respiratory surface will be decreased, and gradually what will happen, your alveoli will die and your lungs will shrink, and you cannot respire no more, and you may die. So it's a chronic disease. That's why it's suggested that avoid smoking. Next case, that is ORD, occupational respiratory disorder. Now this disorder is related to occupation. What is can somebody tell me what is difference between disease and disorder? Can somebody tell me what is difference between disease and disorder? You can write down in the comment section. So occupation means those who are working in grinding industry or stone breaking industry. So if somebody is working in such industries, they have to inhale lot of dust particles, right? They have to inhale lot of dust particles, and this dust inhalation, those dust particles will get inside your lung and will hit your lung surface, and that will cause accumulation of too much amount of fibers inside your lung. And if more fiber is deposited, more fibers are produced inside your lungs, then your alveolar wall will become stiff, will become hard, and that will create inflammation, and your breathing will be problematic, and gradually your lung will die. And some diseases are there, silicosis due to inhalation of silicon particles, asbestosis due to inhalation of asbestos particles, black lung disease, those who are working in collieries or coal mines due to inhalation of too much of coal particles, this occupational respiratory disorder can be occurred. Okay, so here we are done with this chapter. So here we will be solving few questions. So the question is, the partial pressure in millimeter Hg of oxygen and carbon dioxide at alveoli. Okay. Can you answer by writing A, B, C, D? The partial pressure of oxygen and carbon dioxide at alveoli is what? C. Partial pressure of oxygen. Okay, Shubir is saying C. Is it? Partial pressure of carbon dioxide and oxygen. Partial pressure of oxygen is 104 and partial pressure of carbon dioxide is 40. So the answer is option A. Next, which of the following best describes the functional residual capacity? Which of the following describes the functional residual capacity? The total volume of air a person can inhale after a normal expiration, the volume of air that remains in the lungs after normal expiration, the maximum volume of air a person can exhale after a forced inspiration, the maximum volume of air a person can breathe in after forced expiration. Write your answer in the comment section. For this question, the answer will be A, B, C or D. Can you write down in the answer? Okay. For this question, the answer is going to be option B, the volume of air that remains in the lungs after normal expiration. Yes, exactly. Option B. Niharika, Prajwal, Flame Emperor, Subir. Okay, they have given correct answer. Now next question, assertion and reason. The diaphragm plays a crucial role in the process of inhalation. The diaphragm plays a crucial role in the process of inhalation. That is obviously true. When a diaphragm contracts, it flattens and increases the thoracic cavity volume, leading to a decrease in the pressure. Okay. Now, can you answer this particular question? Prajwal, Flame Emperor, Niharika, Shubir. Can you answer this question? when the diaphragm contracts, the reason is saying it flattens, obviously it flattens it and increases the thoracic cavity volume, leading to the decrease in pressure. That's also true. Yes, so I got the answer from Subir. Both the assertion, yes, exactly, both the assertion and reason are true, and reason is the correct explanation for assertion. Okay, so the next chapter I'll be starting, that is body fluid and circulation. So I'll be coming back just within 5 minutes. Then after that, we are going to start the chapter, that is body fluids and circulation. Just 5 minutes. Okay, so welcome back everyone to this session again, and now the chapter I'll be completing, that is body fluids and circulation. And particularly you just write down and tell me in the comment section what are the topics from this chapter particularly want to be get covered. What are the topics you face generally difficulties from this particular chapter? Write down in the comment section. So I'll try to specifically cover those topics only. Okay. So basically, if we say that uh, these are the needs of our transportation or circulatory system for our body, that is delivery of nutrients and oxygen to the tissue, to the cells, and secondly the removal of harmful excretory products as well as carbon dioxide from the tissues, right. So these two are the major function of our transportation system, and along with them our body fluid also transporting hormones and a few other important components of our body, right. Okay. So if you observe the animal kingdom, few animals will be showing water driven circulation. They don't really have blood or something called body fluids, so that's why they have to rely upon water only. So if we talk about sponges, if we talk about cylinderates like hydra, jellyfish, they don't have any kind of body fluid. So with the help of water only, they are performing function of respiration, excretion and circulation. Similar thing goes for the animals with water vascular system like echinoderms as well, right. They also have water driven circulation only. Now the animals, few animals with special body fluid, either you can call the body fluid as blood or you can call it as lymph. Okay. So for more complex organisms, we can see the presence of blood and lymph as their circulatory fluid. Okay. So first we will be talking about blood. So we know that blood is a special connective tissue consisting of a fluid matrix that we call plasma. That fluid matrix is called plasma. Okay, it's already written here. And formed elements. And what do you mean by formed elements? The cells present inside, that is WBC, RBC, and the broken pieces of cells called platelets, right. Okay. One more thing is written here. Blood is considered as false connective tissue. Blood is considered as afibrous connective tissue. Why blood is called as false connective tissue? Because this fluid matrix plasma, the fluid matrix plasma of blood is not produced by the cells of its own. Rather, plasma, the fluid matrix of blood, is produced by some other organs like liver and spleen. That's why it's a connective tissue where matrix is produced by some other cells or other organs or other tissue. That's why we are calling it as false connective tissue. Okay. The process of formation of blood is called as hemopoiesis or hematopoiesis. And you know that where inside our body hemopoiesis is happening? The prime place for hemopoiesis, where inside our body blood formation is happening, that is our red bone marrow, mainly red bone marrow. Okay. Along with that, liver, spleen and other lymphoid organs are responsible for blood formation. Okay. And the hormones you require for hematopoiesis or hemopoiesis, primarily erythropoietin, which is produced by your kidneys. Then your cortisol can stimulate your bone marrows to produce blood. Thyroid hormones can stimulate your bone marrows to produce blood. And also, as I said, that testosterone or androgens, the male sex hormones, can stimulate our bone marrows for the formation of blood. Okay. Now, average adult human being contain 5 liter of blood, or 5 to 5.5 liter of blood inside our body, right? And you know that male is the volume of blood in average male is little bit half or 1 liter higher than the volume of blood in female. Okay. Now we need to talk about different components of blood. If you take 100 ml of blood from your body and keep in undisturbed condition inside the test tube, then what you can observe that 45% of the volume is settled down, the reddish brown portion, that is called as formed elements, and the colorless fluid that is floating over, that is containing the 55% of volume, that is called your plasma. So here we are going to talk about composition of blood. We'll be telling about composition of blood. Okay. So the matrix, the fluid matrix that contains 55%, that takes 55% volume of your blood, that is plasma. And what's plasma? A straw colored viscous fluid. Basically this is the matrix of your blood. That means, what do you mean by matrix? Matrix means ground substance, the medium which is responsible for holding the cells. So it's plasma which is holding the RBC, WBC and platelets. Now what are the different components of plasma? 90 to 92% is only water, and 6 to 8% or 8 to 10%, that is proteins and other solid components. So proteins are the major component. What are the proteins present into your plasma? Fibrinogen. Fibrinogen is acting as a blood clotting factor. You have 13 clotting factors present into your plasma. In the highest concentration you will be finding, that is of fibrinogen protein. And remember, this is the largest protein present into your plasma, largest plasma protein of your blood, fibrinogen. Albumin, smallest but most abundant plasma protein, smallest in the range of molecular weight. The smallest part plasma protein is albumin, but most abundant, 4% of plasma protein, that is albumin only, right? Out of this 6 to 8%, 4%, 50% of the plasma protein is occupied by the albumin molecules, and they help us to maintain water and salt balance of our kidney. That's why it's written here, they help us to maintain osmotic balance or osmotic control. Third plasma protein, that is globulin. Okay, globulin proteins can be of different type: alpha globulins, beta globulins and gamma globulins. Okay, you don't have to talk about alpha and beta globulins because gamma globulins are given in your syllabus and they are very much important for you. Gamma globulins are nothing but immunoglobulins or antibodies. They are IgG, IgA, IgM, IgD and IgE, right? So they help you for our defense mechanism to build our immune system, right? Okay. The mineralic components of our blood, they are sodium, chloride, magnesium, bicarbonate, calcium and other nutrients like glucose, amino acid, lipids are also present into transit form. Now it's very important line of NCERT. What do you mean by transit form? Glucose, amino acid, lipids are present in transit form means the concentration will be fluctuating. I mean, yesterday if I measure blood glucose level, okay, more or less the concentration will be similar, but the molecules of glucose will be replaced today or tomorrow or day after tomorrow. Every day new molecules of glucose or lipids are formed or present into your blood. That's why it's written as transit form, with little bit fluctuation in their concentration. Okay. Now one more important thing, if you eliminate all the 13 clotting factors, how many clotting factors are there into your plasma? 13. Previously it was considered as 14, but right now we study as 13 clotting factors are there. And if you eliminate all the clotting factors from your plasma, the colorless liquid you are gonna get, that is called as serum. Again, a very important question can be asked from this. Okay, now let's talk about formed elements. Now, what is the volume of formed elements present into blood? That is 45%. Now, why these RBC, WBC platelets are called formed elements? Okay, here is the concept that formed elements means RBC, WBC platelets, they are formed inside the bone marrows and then released into the bloodstream. Okay, now these formed elements are can be divided into two groups, that is corpuscles and platelets. What do you mean by corpuscles? RBC, WBC, red blood corpuscles, white blood corpuscles. RBC commonly called as erythrocyte, WBC called as leukocytes because of their color. Erythros means red and leukos means white. Okay, the number of RBC per cubic millimeter blood, milliliter blood, that is 5 to 5.5 million, that means near about 50 lakh per millimeter blood, per cubic millimeter or per deciliter blood. Okay, per deciliter blood or per cubic millimeter blood contains 6K to 8K, 6000 to 8000 WBC. And if we talk about platelets, as because they help us in blood clotting, we call them thrombocytes. Thrombosis is the process of blood clotting, and their count in per cubic millimeter blood or per deciliter blood is 1 lakh to 3.5 lakhs of platelets. Okay, so this is how different formed elements can be observed into a blood smear, right? So these are RBC biconcave shaped, because the matured one, they don't have what? They don't have nucleus, mitochondria, Golgi complex, endoplasmic reticulum. Most of the important cell organelles are vanished when the RBC is matured into our body, right? These are the WBCs. This is the acidophil. How you can identify the acidophil? When you are observing under the microscope, they can take only, you can stain them, you can color them with the help of acidic stain only, and the stain is required, that is called eosin. That's why acidophils are also called as eosinophil. And this is the identification mark, headphone shaped multi-lobed nucleus with granules. See, acidophils, basophils and neutrophils, these are called granulocytes of WBC because of presence of this enzyme filled granules inside their cytoplasmic component. Okay. Now, this is basophil WBC, and how you can identify the most rarest WBC into our body, and S-shaped trilobed nucleus can be observed into basophil molecule. And neutrophil, the prime phagocytic molecule, they can move like amoeba. So these neutrophils are the prime amoeboid cells of your blood and the most abundant WBC, right? How you can identify them? 3 to 4 lobes of nucleus, and when you are staining these neutrophils, either you can stain them with basic stains or basic colors or acidic stain, but for basophil molecules, they will be only stained or colored with basic color, basic stain only. Okay. Now, let's talk about some agranulocyte WBC. There is no granules present into cytoplasm. Cytoplasm looks quite clear. If you find kidney shaped nucleus inside, then it's monocyte WBC. Monocyte WBC is another amoeboid movement showing WBC for us. And if you are finding this cartwheel shaped nucleus, that's their lymphocytes, and you know that there are T lymphocytes and B lymphocytes are there. Monocytes are the largest among WBC. Lymphocytes are smallest among WBC, and sometimes these monocytes, if it's coming into the tissue from bloodstream, it will change the shape and the structure a bit, and the large monocyte is called macrophage, which is a killer machine for pathogens. So macrophage and monocyte basically are the similar cell. When monocytes are present into blood, we are naming them as monocytes. When this is coming out of bloodstream or blood vessel and entering into your tissue to to support your neutrophils while your body is getting attacked, then we call them as macrophage, large cell that is responsible for eating the debris, eating the bacterial cells, right? And obviously the broken pieces, not the broken heart like you, broken cells, broken pieces of megakaryocyte cells, platelets. Remember, if you are asked that which blood component, which formed elements are not actually true cells, they are broken parts actually. So these are platelets. The meaning of platelet means broken piece. Okay. So first we will talk about RBC. So the process of formation of RBC is called erythropoiesis. And where it's happening, RBM, red bone marrow, inside your red bone marrow, RBC is formed, right? What is the number of RBC? Already we have spoken about that, 5 to 5.5 million per cubic millimeter blood. Average lifespan is 120 days. After 120 days, your RBC is gonna expire and a new set of RBC will be replacing the old set of RBC. And do you know that which organ is called as the graveyard of RBC? Can you write down in the comment section? Upon expiry, which organ will be trapping those expired RBC and killing them? Can you write down in the comment section which organ is called as graveyard of RBC? Can you write down which organ is called as graveyard of RBC? So it's written here as spleen, the graveyard of RBC, because the expired RBCs are trapped inside this spleen and killed. Okay. So you know that 100 ml of blood contains 12 to 16 grams of hemoglobins, and which is the location of hemoglobin into our body inside the RBC. And in order to accommodate so like high volume of hemoglobin, like if you consider the space inside the RBC, 36% volume is occupied by hemoglobin molecules only, and that is the reason in order to carry these hemoglobin molecules, matured RBCs will be kicking out, literally kicking out their nucleus and mitochondria and other important cell organelles, right? Okay. Now WBC, the process of WBC formation is called as leukopoiesis. Similarly, the WBC formation is also taking place into RBM, red bone marrow inside our bones. The number is nearby 6K to 8K per cubic millimeter blood, 6,000 to 8,000. Average lifespan of WBC can vary from 10 hours to 10 days depending upon which WBC is this, right? Okay. So WBC can be overall divided into two groups, granulocyte and agranulocyte. If WBC is having granules and multi-lobed nucleus, then it's called granulocyte. If WBC is having single lobed nucleus with no granule inside the cytoplasm, they are called as agranulocyte. Okay. Again, granulocytes are of three types. Again, granulocyte of three types: basophil, eosinophil and neutrophils. Okay, basophil, the rarest cells among our blood, and you can find it here. Only 0.5 to 1% of blood cells of WBCs are basophils, 2 to 3% of WBCs are eosinophils, and 60 to 65% of cells are neutrophils. Already I have discussed what is the reason behind the name of basophil, eosinophil and neutrophil, and obviously why there is so much abundancy of neutrophils inside our bloodstream, as because they are the prime phagocytic cell. Whenever you are getting any kind of infection into your body, is the phagocytic cell, is the neutrophils, they are attacking those spammers. Like one of the spammer is trying to spam in the chat section. So similarly, when the microbial spammers, the infectious agent try to spam or infect your body, is the duty of neutrophils to sustain and to fight those attacks. Yes. Now, if we talk about a granulocyte, there are two types: monocytes and lymphocytes. The percentage of monocytes present into our bloodstream, they are 6 to 8%, and the percentage of lymphocytes 20 to 25%. So overall, if you are asked which is the most abundant WBC, it's neutrophil. Which is the rarest, least abundant WBC, basophil. Second most abundant WBC, lymphocyte. Second most abundant. Third most abundant, monocyte, then eosinophil, and then basophil. Okay, so we are done with WBCs. Next, let's talk about platelets. Let's talk about platelets. Yes, these are broken pieces, these are broken parts of cell. See what happens inside our bone marrow, there will be a process happening every time, that's called thrombopoiesis. That's called thrombopoiesis. And how many platelets are produced? 1.5 to 3.5 lakhs of platelets are produced in per cubic millimeter blood, and the platelets are having average lifespan of 7 to 10 days, and this is the cell from which platelets are produced, and the cell is called as megakaryocyte. You can see, you can observe it here, the cell is with a large nucleus. That's why a large karyon, the megakaryocyte. During the time of maturation, this megakaryocyte will be broken down into smaller pieces, and these broken pieces are called as platelets, and the literal meaning of the platelet is broken pieces of megakaryocyte or broken pieces of cell. That's why it can be said that platelets are not true cells. Platelets are not true cells. Why they are called as thrombocyte? Okay, it's because platelets can release a variety of substances, most of which are involved in the coagulation or clotting of blood. A reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. So, a very important factor required for us, if you are getting a wound and blood is oozing out from your blood vessels, so the platelets gonna come to that place and will be secreting platelet factor or thromboplastin produced by platelets, and they are gonna initiate the blood clotting mechanism into your body. Okay, now the most interesting part of this chapter, that is blood grouping. And we know that according to the Karl Landsteiner, yes, is the pioneer of blood grouping. According to the Karl Landsteiner, the most common blood grouping system can be observed among us, that is called ABO system. And later on one more system was introduced, that is RH system. So today we will be talking about this ABO system and RH system. Now remember, depending upon what blood groups are divided, we have to check for the RBC and the plasma. See, on the surface of RBC, you will be finding antigens, special glycoprotein molecules, and some proteins are present into our plasma. We call them antibodies. So by the presence of these antigenic molecules and antibodies into plasma, remember, what is the place of antigens on the surface of RBC? Place of antibodies in the plasma. We have to decide which blood group is this. Now let's elaborate this a bit. So we have taken 4 different RBCs from 4 different people. These all are RBC. By studying RBC only, you can decide the blood group, right? So if you got for one person you got RBC and the person is having A antigen on the surface, so the blood group of that person will be said as A blood group. You got another person, you have taken the RBC and there you got the antigenic substance present on the surface of that RBC, that is called B type of antigen, and overall the blood group will be said as B blood group. You got another person where the RBC surface is displaying one B antigen and one A antigen. The RBC surface is displaying B antigen as well as A antigen, so the blood group gonna be AB. The blood group gonna be AB for that person. Now you got another person, you are not finding neither of them, nor A antigen, neither B antigen, so the blood group of the person will be O. So that's why I was saying that based upon the presence of antigens on RBC surface, we can differentiate the blood groups as A, B, AB and O. And if you look for the antibodies present into the plasma, if you look for the antibodies present into the plasma for the person A, this is the antigen. These all are what? These all are antigens. These all are antigens. So the plasma will have the antibodies. The plasma will have the antibodies. For the person which is having blood group A, the antibody of plasma will be anti-B. The person with blood group B, the antibody in the plasma will be anti-A. The person with AB blood group, you won't gonna get any of the antibodies into the plasma, and for the person with blood group O, you are gonna get both the antibodies named anti-A as well as anti-B. And some of you are knowing this fact that this AB blood group, the person with AB blood group is said that universal recipient, and the person with O blood group is said that they are universal donor. What is the reason? Why this person with AB blood group can receive blood from everyone? because they don't have any antibodies into their plasma. Who are the villains? Antibodies are the villains. Antibodies are the villains. If you have antibody inside your body, then this antibody is going to attack the antigens. But for AB blood group person, there is no antibodies in their plasma. So if they are taking blood from a person with blood group O or blood group AB or blood group B or blood group A, there is no antibody which can detect antigen. That's why there will be no coagulation activities. So that's why always they are safe. They can take any blood. That's why we call them as universal recipient. But why O blood group persons are called as universal donor? See, they can receive blood only from the person with O blood group because they have both the antibodies, that is anti-A and anti-B, but they don't have any antigen displayed on their RBC surface. So they can donate blood to every other person, and the antibody present into their body cannot detect any of the antigens. So that's why it's the advantage of this particular blood that if you have blood group A, you can take blood from the person with O blood group. If your blood group is B, you can take blood from the person with O blood group. If you are AB, similarly you can take blood from this person, and if you are blood group O, you can take blood group. That's why we are telling them universal donor. Okay. Now we'll talk about RH antigen. Now we'll talk about RH system. We are done with ABO system. Now we'll talk about RH system. Now Karl Landsteiner and his group, when they were searching for the antigen present on the surface of RBC, along with A antigen and B antigen, they got another antigen called as R antigen. Along with A antigen and B antigen, they discovered another antigen, especially when they were checking the blood or RBC of rhesus monkey. They discovered this particular antigen on the RBC of rhesus monkey, and then a similar kind of antigen was found in the RBC, on the surface of RBC of human. So that's why we have named it as RH antigen, by tributing that particular discovery of this kind of antigen which is present on the surface of RBC of rhesus monkey. So RH for rhesus, right? So another antigen, the RH antigen, similar to one present in rhesus monkeys, hence its name as RH, is also observed on the surface of RBCs of majority, nearly 80% of humans. So such individuals are called as RH positive, and those in whom this antigen is absent are called RH negative. So as simple as that. If I take your RBC and if I find this RH antigen, then I will be saying your blood is positive group, and if I am unable to find this RH antigen, then I will be saying your blood is negative group, right. So if this person is having B antigen and RH antigen, then I will be saying the blood group will be B positive, right. If a person is having A antigen here and RH antigen here, then I will be saying the person is having A positive blood group. If the person is having A antigen as well as B antigen and RH antigen, then I will be saying the person is having AB positive. So this is how blood group is determined for positive person and negative person. Now see how blood group is determined. So suppose I have taken blood from your body. I have taken blood from your body to determine your blood group. So what I will do, I will take 3 slides and I will put 3 drops of your blood into these 3 slides, on these 3 slides. Okay. Now what I will check, I will first put few drops of anti-A on this slide. I will put few drops of anti-B on this slide, and I will put few drops of anti-RH on this slide. Now suppose I have seen that the blood of second slide, that is coagulated, and blood of third slide is also coagulated. Now can you comment in the comment section that the blood group is what? I have taken blood from a person and I have distributed the blood as a 3 drops into 3 different slides, slide A, slide X, slide Y, slide Z, and I have put what? 1 drop of anti-A, I have put 1 drop of anti-B, I have put 1 drop of anti-RH, and the blood is coagulated for anti-B slide and anti-RH slide. What is the blood group of this particular person? Can you comment? Okay. See, when there will be coagulation reaction, here anti-B is reacted here. That means that blood is containing, B antigen. Only B antigen can react with anti-B. Here you have put anti-A, but there is no coagulation, that means there is no presence of A antigen. Here upon putting anti-RH, the blood is coagulated, that means here you will be finding presence of RH antigen. So that means B is present here and RH is present here. The blood group of this person is B positive. Right. If you have observed that slide X is coagulated, slide Y is coagulated and slide Z is also coagulated, then the blood group will be AB positive. If none of the slides are coagulated, then the blood group will be O negative. If X and Y slides are coagulated but Z is not coagulated, then the blood group will be AB negative. So this kind of question can be asked from this particular topic. Okay. Now one question is here, which one of the following statement is incorrect? So let's check the statements. A person of O blood group has anti-A and anti-B antibodies in his blood plasma. That is correct. A person of B blood group cannot donate to blood to a person of A blood group. This statement is also correct. Question 3, blood group is designated, blood group is designated on the basis of the presence of antibodies in the blood plasma. Blood group is designated on the basis of the presence of antigens on the RBC. So this is our answer, because this is a wrong statement. The fourth statement, a person of AB blood group is universal recipient. This is also true fact. So we got the wrong statement, option 3. Next, we'll talk about hemolytic disease of newborn, or name of the disease is erythroblastosis fetalis. Now, why the disease is named so, erythroblastosis fetalis? Okay, first of all, it is observed in fetus, right? The embryonic stage of the baby inside the mother's womb. That's called fetus. Okay. Now, what do you mean by erythroblastosis? Erythroblastosis is a condition where your mature WBCs are gonna destroyed by your immune system or by some immune system, and your body is only having immature RBC. So, the immature RBCs can be called as erythroblast, and there is only erythroblast into your body, there is no trace of matured RBC. This condition is called as erythroblastosis. Do not think that erythroblastosis means erythrocytes are getting blasted. That is not the fact, right? Here the fact is erythroblasts are the only cell present into the body of baby. There is no trace of mature RBC. Then where are those mature RBCs? They are killed by the antibodies. They are killed by the antibodies. Now let's check how it happens. Suppose if the blood group of father is A positive and the blood group of mother is A negative, then what will happen? Suppose the first baby, the first child born with the similar blood group of father, that is A positive. So this is the case. So during the time of delivery of this first child, the blood will enter from the child to mother body, because rupture of blood vessels will be there during the time of parturition, and as because A positive blood is entering into the mother's blood which is A negative, always remember Rh negative person cannot tolerate blood of Rh positive person, but Rh positive person means blood with Rh positive can tolerate blood of Rh negative. If I am Rh negative, if I am A negative, then I cannot take blood of A positive. But if I am A positive blood, I have A positive blood, happily I can take blood from A negative person. So that is the thing, that is the thing happening here. Mother blood is A negative, and during the parturition of first baby, the baby's blood which is A positive, which is entering into mother's blood circulation, and that's promoting a threat to mother's immune system, and here mother body started producing anti-RH. Mother's body started producing anti-RH in the response of this A positive blood, in the response of the Rh antigen present into the A positive blood. Now what will happen? That will not create any effect or adverse effect on the first child. Already the first child is delivered successfully. Now the problem will appear from second child onwards. Now what will happen? The second child, though separated from the placental barrier, but this anti-RH are produced, which are produced inside the mother body, they can cross this placenta. That is the irony. Though other antibodies cannot cross the placenta, but anti-RH can cross the placental barrier, and now it will start attacking RBC, mature RBC. It will destroy mature RBC. It will attack the liver of that second child, and if there is no RBC, all RBCs are destroyed by this anti-RH, and liver cells are damaged. So the child will be suffering from severe anemia and jaundice, and in worst condition the second child may die, or second child or third child onwards they may die. So this condition is called as erythroblastosis fetalis. Okay, but one thing is there, this condition can be avoided. How? By administering anti-RH antibodies known as ROGAM. So to the mother immediately after the delivery of the first child. So after the parturition of the first child, if you inject ROGAM, into the mother's body, as soon as it will get this RH plus RH antigens, it will attack those RH and will destroy just prior to posing any threat to mother immune system and just prior to formation of anti-RH. So now if you are administering ROGAM or anti-RH antibodies just after the parturition of first baby, the mother cannot produce anti-RH, so there will be no threat exerted to the second child and onward. So that was all about hemolytic disease of a newborn or erythroblastosis fetalis. Okay. Now I'll talk about coagulation of blood. So coagulation blood, when you are getting any wound into your body, then you can see that your blood is coming out, blood is oozing out gradually. But within few minutes, your body's mechanism will stop that process. Your body will seal the damaged portion of the blood vessel by forming some fibers, called fibrin, right. As we know that blood is a fibrous tissue while it's flowing, but when we need to coagulate the blood, when we need to coagulate the blood, we have to form some fibers, and these fibers are called as fibrin. Now let's check how to produce that. See, whenever there is tissue injury into your body, that damaged tissue will start releasing thromboplastin. This thromboplastin, this is one of the precursor molecule for starting the process of blood clotting or blood coagulation. Simultaneously, the blood vessels will also be injured in that place, in that tissue injury place. So that will secrete some components, some chemicals, some chemoattractants, and they will call platelets towards the wound, towards the injured part. And as soon as platelets will be gathering near to that, they will start releasing platelets factor or platelets thromboplastin or endothromboplastine. So two kinds of thromboplastins are produced. First, exothromboplastine, that is produced by the injured tissue, and endothromboplastine, that is produced by the platelets which were fetched where blood vessel is injured. Now here we need calcium ion. In the presence of calcium ion, this tissue thromboplastin and platelet thromboplastin will bind with each other and will convert, that will act as an enzyme, and we call that thrombokinase. In the presence of calcium, tissue thromboplastin and platelet thromboplastin will bind with each other. Suppose this is the tissue thromboplastin and this is the platelet thromboplastin. So they will bind with each other to form thrombokinase. Now this is a catalytic enzyme. Now this thrombokinase will activate one switched off protein that is called prothrombin, and the activated form is called as thrombin. Once thrombin is formed, it will catalyze another reaction. It will switch on another sleeping protein called as fibrinogen, and it will activate as fibrin. Once the fibrins or fibers are produced, once the fiber in the form of fibrins are produced, so what they will do, if this is the blood vessel and this is the leaked portion or injured portion, they will settle here by forming a mesh, and they will trap more and more RBC, and that will create a plaque, that will create a RBC plaque kind of structure, and that will seal the opening, and that is called as blood is clotting there, and within few minutes or within few hours your your platelets factor gonna repair the damaged portion of your blood vessel. So that's what we are telling, that this process as blood clotting or blood coagulation. Next, here we got a question, and the question is, a reduction in the number of platelets can lead to blood coagulation inside the vessels, clotting disorders, excessive loss of blood from the body. Okay, a reduction in the number of platelets will not cause blood coagulation in the vessels, but it will trigger clotting disorder, it will trigger excessive loss of blood from the body. So the answer will be option 4. Now we will talk about lymph. So lymph is often called colorless fluid. Now why lymph is colorless? As because lymph is nothing but filtered blood. Lymph is nothing but filtered blood. Yes, inside your tissue you have some fenestrated blood vessels, means blood vessels with tiny pores. Now while the blood is flowing inside the blood capillaries, few portion of your blood will be filtering out and will start their accumulation into the tissue space, and the tissue space, the fluid is accumulated, is also called as tissue fluid, and the space is called as interstitial space. So the tissue fluid is accumulated into the tissue space, that is called as interstitial space. So that's how lymph is produced, just nothing but filtered blood. Blood is getting filtered out from some specific blood capillaries into the tissue space, and that's why we call tissue fluid the another name of lymph. Okay, now why it's colorless? Because lymph is having similar components of plasma, and we know that plasma is also colorless. Okay, and some lymphocytes. What are lymphocytes? They are special kind of WBC. So a fluid which is present inside the interstitial space of tissue, which is rich in lymphocytes and which has kind of similar plasma of blood, that liquid is called as lymph. Now why do we need lymph into our circulatory system? Lymph is called as helper of blood, right? Blood is confined into our closed circulatory system. Blood is confined into our closed circulatory system. Blood is flowing, suppose this is the circuit through which blood is flowing. Blood is confined in, blood is flowing into a closed system. But we have to send nutrients, gases to the cells of our tissue. So there is no direct connection in between blood and your tissue. So what to do now? So that's why your blood will get filtered out and will form a liquid medium in between the blood vessels and the cells of your tissue, and the liquid present, or liquid which is connecting blood with your tissue, that is called your lymph. That's why lymph is also called as middleman of your body. That's why lymph is also called as middleman of your body, because it's connecting your blood to your cells of your tissue. And now what thing will happen? The nutrients or the oxygen will come first from blood to lymph, and then from lymph to your cells. When cells are releasing carbon dioxide, first the carbon dioxide will come and dissolve into the lymph, and then from lymph to blood. When the blood is trying to deliver the nutrient, so the nutrient will be deposited first into the lymph, and then from lymph the nutrient will be diffusing inside the cells. So that's why basically in our body, blood is a part of closed circulatory system. So directly blood cannot reach to the cells of our tissue. That's why blood has kept an assistant, you can call, that is lymph. So another question here, which of the following is correct? Lymph equal to plasma plus RBC plus WBC, blood equals to plasma plus RBC plus WBC plus platelets, plasma equals to blood minus lymphocytes, or serum equals to blood plus fibrinogens. So can you answer this question by commenting 1, 2, 3 or 4 into the comment section? Yes, Niharika, you have given the right answer. Blood equals to plasma plus RBC, WBC plus platelets. Okay. Now we'll talk about two different modes of circulation, that is open circulatory system and closed circulatory system. And obviously it is considered that open circulation is kind of primitive in compared to the closed circulation. Why open circulation is primitive? See, in open circulation there is transfer of body fluid in between heart and body cavity. Heart is pumping the blood and the blood is directly coming into the body cavity, right? So all the cells or tissues of the body are getting bathed with the body fluid. So the nutrients or the gases or the important transport material are equally distributed to all the cells of their body, those who have open circulation. But in real world, every cells or every tissue, every organ, different organs are requiring different amount of nutrients, different amount of gases. So that's why you cannot deliver similar component to all the tissues. So that's why nature needed a more specified mechanism of circulation, and that's why open circulatory system was upgraded through the process of evolution into closed circulatory system. Now what is the advantage of closed circulatory system then? Heart will be pumping blood into blood vessels, and through different diameters of blood vessel, blood will flow throughout the body, and again the blood will enter into the heart. So here what's happening, blood is not directly opened into the body cavities, rather blood is flowing through blood vessels, and that's the reason different blood vessels will give blood supply into different organs and fulfill the different needs of the body. Who are the animals who have open circulation? Arthropods and mollusks. Arthropods and mollusks, they have open circulatory system, and annelids and chordates. Annelids and chordates like earthworm, leech, and us, we are under chordata. So we have closed circulatory system. Okay. Now let's come to the heart, different kinds of heart present into the vertebrates. So for fish, we can observe two chambered heart. They have one auricle, one ventricle, and that kind of heart is called venous heart. The mode of circulation is called single circulation, because that heart is receiving and pumping only deoxygenated blood, only CO2 rich blood. That's why the heart of two chambered heart of fish, we call them, that circulation is called as single circulation, and the heart is called as venous heart. Okay. Now for amphibia and reptilia, we can observe three chambered heart. For three chambered heart, you can observe two auricles but one ventricle. But due to the presence of only one ventricle, blood is again mixed. Pure and impure blood, oxygenated and deoxygenated blood will be mixed, and that's why for them we can observe incomplete double circulation. For reptiles, and amphibians, we can observe incomplete double circulation or mixed double circulation. Okay. For mammals and birds, and also for crocodile. Mammal, birds and crocodile, we have four chambers in our heart, two auricles and two ventricles, and complete double circulation. Why it's complete double circulation? Because oxygenated blood and deoxygenated blood is completely separated into our heart. Left side is carrying oxygenated blood, right side is carrying deoxygenated blood, and that's why we call these as double circulation. Next, now we'll be elaborating human heart. So our heart is a muscular heart in nature, and we also know that it's called as myogenic heart. Even the heartbeat is generated from inside only, right? We have blood vessels of different diameters. We call them arteries, veins and capillaries, right? Arteries have very thick wall, veins have thin wall, and capillaries have ultra thin wall. And our main body fluid is our blood, which is a kind of specialized fluid connective tissue. Our heart is originated from embryonic mesoderm. That's why it's written as mesodermal as origin. Where inside our body our heart is situated, that is location of our heart, that is in the cardiac notch of the left lung, obviously inside the thoracic cavity. Inside the thoracic cavity, in the place, the space in between two lungs, that is called the notch of the left lung, that is called as cardiac notch. So if this is the left lung and this is the right lung, so the space in between them, that is called as mediastinum. The space in between the two lungs is called mediastinum, and if you observe the left lung, you will be finding this concave notch, that is called as cardiac notch, and here only the apex of your heart, this conic part, that is called apex of your heart, is embedded. So that is the location, and the size is like your clenched fist. Okay. Now let's talk about anatomy. Now I'll be talking about anatomy of our heart. First, we'll start with the outer covering. So the outer covering of our heart is called as pericardium. Now the first layer of your heart, the outermost, below the pericardium, the outermost layer of your heart is called as epicardium. Middle muscular layer of your heart is called as myocardium, and the innermost layer of your heart is called as endocardium. So it's done about the covering of your heart and the layers of your heart. Now we know that we have 4 chambers. So this is the left atrium, LA. This is the right atrium. This is right ventricle. This is left ventricle. So these are the 4 chambers. So now we have to talk about how these chambers are divided from each other. Okay. Now if we check at the middle part, the left side and right side of our heart is divided by septum, means cardiac wall. Now the septum, if you observe here, is thicker in between the ventricles, and it is very thin in between the atrium, right? Very thin in between the atrium. So the septum, which is separating right ventricle to left ventricle, that is called as intraventricular septum. Ventricular septum. The septum which is separating right atrium to left atrium, that can be called as interatrial septum. So 2 septum is there, atrial septum and ventricular septum, and question can be asked which is thicker, which is thinner. Obviously ventricular septum is thicker and atrial septum is thinner, and in between them there is a connector septum which is connecting atrial septum to ventricular septum, that is called atrioventricular septum. What is the difference between atrioventricular septum, atrial septum and ventricular septum? Atrial and ventricular septum, they both are muscular. They are made up of muscle. But atrioventricular septum, that is made up of fiber. So it's fibrous septum. Okay. So that's done, how to separate out, how to divide the left and right side of the heart. Now let's divide right atrium to right ventricle. Okay. So valve is present in between right atrium and right ventricle, that is called as tricuspid valve. It is called as TCV, tricuspid valve, because that is having 3 flap or 3 cusp, and the valve which is separating left atrium to left ventricle, this atrioventricular valve is called as bicuspid valve, BCV, and this bicuspid valve is also known as mitral valve. Why it's called as mitral valve? Because the shape of this valve is somehow is the cap of mitre of charge. That's why it's known as mitral valve. Obviously, it's having 2 flap, 2 cusps. That's why you named it bicuspid valve. Okay. Now, this bicuspid and tricuspid valve, they can be called as AV valve or atrioventricular valve because they are separating atria to ventricles. Now, you need to control the closing and opening. You need to control the closing and opening of bicuspid and tricuspid valves. So, how to control that? So, for that we have some fibrous strips that is called cordy tendineae. They are called cordy tendineae fibrous structure which will be connecting this bicuspid and tricuspid valve with the papillary muscle of the with the wall of ventricle. So, it's cordy tendineae, this fibrous structure which are holding tight this bicuspid and tricuspid valve. One end of this cordy tendineae they are connected with these valves and another end connected with the papillary muscles or columnae corni. So, that is how this opening and closing of bicuspid valve and tricuspid valve are controlled by the force or the stretch created on this cordy tendineae. Okay. Now, a few more valves are present inside our heart. So, before that we will need to we need to talk about the plumbing system, means the vascular system of our heart. So, from left ventricle, the systemic, the largest artery of our body is originated, that is called aorta or systemic artery. Aorta is also called as systemic artery. Here this C shaped or the J shaped arch is called as aortic arch, and from here different branches going towards our limbs, towards our brain. Okay. Now, from right ventricle, another important aorta is originated, and that is taking deoxygenated blood to the lungs, and this T-shaped aorta is called as pulmonary aorta. Pulmonary artery. Pulmonary artery, not aorta. Pulmonary artery. Okay. So, aorta is the systemic artery and pulmonary artery is the exceptional artery which is carrying carbon dioxide rich blood, to the left and right lung for oxygenation purpose. Okay. Now, a prime vein which is open into the superior side of right atrium is called superior vena cava, SVC, which is fetching deoxygenated blood from the upper part of the organs which is situated upper side of the heart, and another prime vein which is coming from the downwards and opening into the right atrium is called inferior vena cava or IVC. These are called as systemic veins. Okay. Now, you will be finding 4 openings into the left atrium where oxygenated blood is deposited from lungs, and the carrier blood vessels are actually veins. These are exceptional veins who are carrying oxygenated blood, and these are called as pulmonary veins. Now you can observe that aorta is somehow connected to this pulmonary artery with the help of a fibrous structure. This structure is actually a ligament kind of structure called as ligamentum arteriosum. That is not important for us. Okay. So, a ligamentum arteriosum is present, which is a fibrous connectivity in between the outer wall of aorta with the pulmonary artery. Okay. Now, we have to check the opening or the beginning of this pulmonary artery and this aorta or systemic artery. So, the opening of both aorta and pulmonary artery will be finding three tricuspid valves, three semilunar valves. Why semilunar valves? Because if you zoom out them, the opening the initial opening of this aorta, then you will be finding moon shaped, half moon shaped valves which is guarding the opening, initial opening of aorta and pulmonary artery. That's why we have named them as semilunar valves. Luna means moon. Yes, Surya, very good evening to you. Okay. Now we'll talk about the conducting system of our heart. Conducting system means we know that the middle layer of our heart, that is called myocardium, that is made up of cardiac muscles. Now, few modified version of cardiac muscles are present into different zone of our heart. That's is making it possible to transport or to carry uh, chemical electrochemical signal throughout the muscles of heart, and that is making the heart to contract and relax. So we know that our heart is beating, and this is called auto excitability, and that's why we call it our heart is myogenic in nature. So why our heart is myogenic in nature? Because it has some modified cardiac muscle which can produce heartbeat, and that is due to the presence of special kind of cardiac muscle called as nodal tissue. Nodal tissue are nothing but modified cardiac muscles. Okay. Now the prime nodal tissue, the prime center from where this heartbeat is generated, the depolarization, the electrochemical signal is generated, is present at the outer left corner of your right ventricle, of your right atrium, of our right atrium. And as because the depolarization generated by this particular part is responsible for heartbeat, that's called as pacemaker, and we know that pacemaker is also called as sinoatrial node or SAN. Basically the SAN is responsible for creating this heartbeat. Now this SAN will be throwing the depolarization through internodal fibers and temporarily it is stored into a battery kind of structure called as spacer that is present right at the opposite corner of the SAN, that is called atrioventricular node, at the junction of right atrium and right ventricle. That's why it's called as atrioventricular node. Okay. Now for a time being, this atrioventricular node will be pausing, and just within few seconds, within few milliseconds, it will be shooting the beat or the depolarization inside the septum present in between the right ventricle and the left ventricle through the ventricular septum. So the fibers which are coming out from atrioventricular node, we call them AV bundle. You can call them as bundle of His. Now, this bundle of His or AV bundle will be bifurcated as a left AV bundle and right AV bundle. Left AV bundle and right AV bundle, and gradually the left AV bundle will enter into the ventricular musculature of left side and the right AV bundle will enter into the ventricular musculature of right side, and further we can observe branching of these AV bundles or bundle of His, and we name them Purkinje fiber. So that's forming a closed network inside our cardiac muscle or heart musculature, that the impulse is produced when it's produced by SAN can travel throughout the cardiac muscle, and that will help the heart to contract and relax. So what is the pathway? It's starting from SAN. SAN will send the depolarization into AV node or AVN. From AVN, it will come into the AVB, AV bundle. Then AV bundle, from AV bundle to left and right AV bundle or left and right bundle of His, that will enter into the ventricular muscle, and that will be branched into Purkinje fiber. So this is the circuit of nodal tissue into our heart. Okay. Now a question is there: all vertebrate possess a dash. Fishes have a dash chambered heart with atrium and ventricles. Amphibians and reptiles have a dash chambered heart. Birds and mammals have a dash chambered of heart. Choose the correct answer. So you can comment your answer just below in the comment section. Okay. So Niharika is saying it's option 2. Obviously vertebrate possess a muscular heart. Fishes with 2 chambered, reptilians 3 chambered, and mammals with 4 chambered heart. Okay. Now we'll talk about cardiac cycle. So what is cardiac cycle? Heart is beating to pump blood throughout the body, and the continuous contraction and relaxation is called as cardiac cycle. So it's sequential repetitive contraction and relaxation of chambers of heart, that is simply called as cardiac cycle, and it's divided into two segment. One is systole and one is diastole. Now systole means contraction and diastole means relaxation. Why heart is performing systole? Systole means blood you just get out. Systole means to pump the blood from heart throughout the body, and diastole means heart is making place to receive blood. It's welcoming blood inside the chamber. So diastole means when heart receiving blood, then it will be performing diastole. When heart is pumping the blood, then it will be performing systole. Okay. Now overall, if we check the cardiac cycle, it has major three phenomena. For one cardiac cycle, for one systole and diastole, for one cardiac cycle, the timing is 0.8 second, right? Now this 0.8 second time span is subdivided into different parts. So if this is a clock and if you start ticking, if this is 0.0 second, from here to here if you are coming back, then that will be 0.8 second. From here to here, one round the clock, one round around the clock, that is 0.0 to 0.8 second. Okay. Now the first phenomena as described in NCERT, that is called as joint diastole. Joint diastole, that means where atria and ventricle both are in diastolic mode, and that duration is 0.4 second, that is half of the cycle is occupied by joint diastole. Now just after completion of joint diastole, it will be atrial systole. It will be atrial systole, and it's the fastest and shortest phenomena. The duration is only 0.1 second. So the remaining 0.3 second, that is nothing but ventricular systole. So as per NCERT, these three are the major steps of cardiac cycle. Getting started with joint diastole, then after joint diastole we can observe atrial systole, after atrial systole we can observe ventricular systole, and again from here we can observe joint diastole. So these three major phenomena we have to study one by one. Okay. So first, heart is in joint diastolic mode. So joint diastole means atria is also relaxing, both the atria and both the ventricles are also relaxing. Arrow outward means it's relaxing. Okay. All the valves are closed, bicuspid and tricuspid valves are closed. Semilunar valves of both the atria of aorta and pulmonary artery, they are also closed, and that's why we can observe equal pressure, right, distributed in all the chambers. Now what's happening here, this atria is gaining blood, when it's diastole, blood is gradually pouring inside the atria. The atria is getting filled with blood, and that will create pressure, and there will be opening of bicuspid valve and tricuspid valve, and gradually the blood will perform a free fall from atria to ventricle, that is called as passive filling. So, during the time of joint diastole, suddenly what happens, bicuspid and tricuspid valve opens and blood will fall from atria to ventricle due to the gravity, and no one is pressurizing the blood to come from atria to ventricle. That's why the process is called as passive filling, because there is no utilization. You don't need to utilize any energy to pump blood from atria to ventricle. Freely blood is coming from atria to ventricle. Okay. And how much blood is coming during the time of passive filling? 70% of blood, 70% of blood is coming during the time of passive filling from atria to ventricle. Now 30% volume of ventricle is remaining. Okay. Now after that, what will happen? Atrial systole will start, and that will be triggered by switching on the SAN. Now SAN will start firing depolarization. SAN will start firing depolarization, and suddenly what will happen? Atria will start squeezing. Now atria will go into systole mode. Atria will go into a contraction mode. So forcefully, it's time, forcefully atria is sending the remaining blood into the ventricle, and how much blood is sent forcefully? That is 30% of the blood is sent forcefully during the time of atrial systole. And as because atrial systole is an active process, muscle contraction of atrial wall, it's an active process, so this filling of blood is called as active filling. So we are done with atrial systole, and the timing is only 0.1 second. Within 1 second, atria will squeeze and it will send the remaining blood completely into the ventricle. Now after atrial systole, ventricle is completely filled, right? Now we will be moving forward to the ventricular systole. See, now what will happen in the beginning of ventricular systole? Ventricular chambers are 100% filled, and what is the capacity of blood? Inside each ventricle, near about 120 ml of blood is present, and that is exerting enormous amount of pressure into the ventricular wall, and that is creating a possibility due to the excess pressure created by the ventricles, because ventricles are going for systole, ventricles are going for contraction, it's creating enormous pressure. There is a chance the blood can flow back to the atrial chamber. So you have to stop that. So how to stop that? So at the beginning of ventricular systole, you have to close bicuspid valve and tricuspid valve, right? You have to close bicuspid valve and tricuspid valve, and that's why during the time of ventricular systole, heart will create its first sound due to the closure of bicuspid and tricuspid valve. During the time of closure of AV valve, AVV means bicuspid valve and tricuspid valve, heart will produce its first sound called lub at the beginning of ventricular systole. So what will happen after ventricular systole? Ventricle will forcefully send the blood into the aorta and pulmonary artery by opening the semilunar valves. So during the time of ventricular systole, one more thing can be observed: opening of semilunar valves. And Now the blood is pumped into the arteries. Okay. As soon as the blood is pumped into the arteries, the pressure will fall inside the ventricular chamber. Now what will happen? There is a chance of flow back. There is a chance of flow back, and at this time you have to close the semilunar valves just prior to ventricular diastole. Now ventricle is going for diastole. Now ventricle is going for diastole because already it has pumped the blood into the atria and pulmonary artery. Now gradually the pressure is falling below, right? Pressure is falling below 80 mmHg. So ventricular systole is completed. Ventricular diastole has begun, and what will happen now? This atrial pressure, now atria is into diastole, atria is gaining blood. So this blood will be creating pressure on bicuspid and tricuspid valve that will gradually promote opening of bicuspid and tricuspid valve, and that gradual opening of bicuspid and tricuspid valve, again it will be from ventricular diastole, it will come into the JD, means joint diastole. During the time of joint diastole, what is happening? Atria is sending blood into the ventricle, and suddenly this bicuspid and tricuspid valves are closed just at the end of ventricular diastole, and that will create the second heart sound called dub. Okay. Now some basic calculation in regarding this cardiac cycle. So what is heart rate? So heart rate means in a minute how many times the heart is beating, that is the heart rate. It is 70 to 72 times, and the reason behind this heart rate is the SA node can shoot 70 depolarization per minute. So that is actually the heart rate. In cardiac cycle, the duration for one cardiac cycle is 0.8 second, and stroke volume means when a heart is doing ventricular systole, each ventricle is pumping 70 ml blood, that is the stroke volume. And utilizing the heart rate and stroke volume, we can calculate cardiac output. So cardiac output is nothing but heart rate multiplied by stroke volume, 70 multiplied by 72. So cardiac output somehow it will be nearby 5000 ml. Okay. Now we will talk about ECG. So what is ECG? ECG is nothing but graphical representation of electrical activity of heart during cardiac cycle. Okay. Now we got two terms. One is electrocardiograph and one is electrocardiogram. Electrocardiograph is the actual name of that machine. Name of the machine is called as, this machine is called as electrocardiograph, and this graph we can fetch from this machine, the graphical representation, that is actually electrocardiogram. So please you have to remember these two terminologies. What is difference between electrocardiograph? Here graph is actual machine and gram is the actual graphical representation. Okay. Now this is how electrocardiogram is taken during the time of performing this ECG. We need some help of electrical leads or electrodes, and three major electrodes are required. One electrode into the wrist of right arm, one electrode the wrist of left arm, and another electrode the left ankle. So that is basically creating a triangular electric field, and as because the apex of heart is kind of triangle, and by measuring the potential difference when the depolarization is thriving inside your cardiac muscle from SA node to AV node, from AV node to AV bundle and the Purkinje fiber, then these nodes, these leads or these electrodes will be calculating the potential difference of different zone and will be creating this sort of graph. So what is the meaning of different waves, different this positive jump towards positive zone or jump towards negative zone? So the when we are studying this ECG graph, if we are talking about the straight line, this is called as isoelectric line. When the graph is jumping upwards, that is called positive deflection. When the graph is jumping downwards, that is called negative deflection. And if we observe, we will be finding P as a positive deflection, Q as a negative deflection, R as a positive deflection, S as a negative deflection, and T as a positive deflection. So the ECG graph is represented as PQ RST. Now here we need to talk about P wave, QST wave and T. So P wave means depolarization of atria. What do you mean by depolarization of atria? Positive ions are thriving inside the muscles of atrial wall, and the consequence of depolarization of atria will be systole of atria. First, there will be depolarization of atria, charges will flow through the muscles of atria, and that will cause atria to systole, atria to pump blood into ventricles. Now there is a this Q straight line is called as isoelectric line. That means there is no overall electrical activity is happening. Now from QRS, this will be forming a QRS complex. This will be forming a QRS complex. This huge wave is called as QRS complex, and this is denotation of ventricular depolarization. QRS means ventricular depolarization. That means as a consequence of ventricular depolarization, there will be ventricular systole. Ventricles are pumping blood throughout the body as a consequence of this formation of QRS wave or QRS complex. Now here this T wave means now repolarization of ventricle. Repolarization of ventricle. Now upon completion of this repolarization of ventricular T wave, you can observe ventricle is again going into diastole. Now remember one more important thing, depolarization is not equals to systole and repolarization is not equal to diastole. It's very important concept to remember. Rather, we can say that systole is a consequence of depolarization and diastole is a consequence of repolarization. First there will be depolarization, then there will be systole. First there will be repolarization, then there will be diastole. That means simultaneously they are not happening. P wave doesn't define arterial systole. QRS complex doesn't define ventricular systole, and T wave doesn't define ventricular diastole, right? So somewhere first there will be P wave, somewhere at the end part of P wave you can observe ventricle has atria has started systoling. Okay, just at this point of the beginning of Q wave, you cannot observe ventricle is systoling. Somewhere at the middle part of the QRS complex, you can observe ventricle started systoling, ventricle started contracting. Okay, at the beginning of T wave where repolarization has started, you cannot observe ventricle is relaxing, ventricle is diastoling. Rather, at the end, somewhere at the near to the end part of this T wave, you can observe ventricle has started relaxing or ventricle diastole, ventricular diastole is started. So that's why it's written in NCERT. T wave denotes ending of ventricular systole. Ventricular systole will start from here and ventricular systole will end here. Okay, that's why I have written here depolarization is not equals to systole, repolarization is not equals to diastole. First you will feel hungry, then you will start eating your food. First your heart muscles will perform depolarization, then systole will start. First your cardiac muscle will perform repolarization, then diastole will start. So it's like that. Now if we talk about double circulation, we know that oxygenated blood is coming from lungs to heart with the help of pulmonary veins, and from left ventricle is the aorta which is circulating this oxygenated blood throughout the organs of our body. Okay. Now from organ we are getting deoxygenated blood. Now this deoxygenated blood is collected by vena cavas. So all the vena cavas are collecting deoxygenated blood and depositing into the right atrium. The deoxygenated blood is coming from right atrium to right ventricle, and from right ventricle this is the duty of pulmonary artery to take back the deoxygenated blood to the lungs for oxygenation. So if you observe this passage, the connection, the vascular connection between lung and heart, that is called pulmonary circuit. The vascular connection between lung to heart, it is called pulmonary circuit, and the vascular connection between heart to organs or system, that is called systemic circuit. Systemic circuit is getting started from this left ventricle to aorta, aorta to systems or organs, organs to vena cavas, and vena cavas to right atrium. That's about systemic circuit. Now what's pulmonary circuit? It is getting started from right ventricle through pulmonary artery coming into the lungs, and from lungs it's the pulmonary veins fetching the oxygenated blood into the left atrium. So from right ventricle to left atrium, this channel is part of pulmonary circuit, and from left ventricle to right atrium, this channel is the part of systemic circuit. So that is the reason we can observe separation of oxygenated blood and deoxygenated blood in both the side of heart. The left side is mainly responsible for carrying oxygenated blood, receiving and pumping oxygenated blood. The right side of the heart is mainly responsible for receiving and pumping deoxygenated blood, and that is actual meaning of double circulation. Now we will be talking about one more system, that is hepatic portal system. What do you mean by hepatic portal system? What is portal system? What is port? See, we know that for all the organs, if this is the organ of your body, that will release some veins to carry oxygenated, deoxygenated blood, and these veins or the venules will be connected to the prime vein of your body or supreme vein of your body, that is called as vena cava, right. So, this is the normal thing what is happening into our body. From organs, deoxygenated blood will be carried by veins, and those veins will be connected at the prime vein or the main vein of our body called vena cava. This is the normal case. This is not a portal system. But in our body, if such thing occurs, you have organ A and organ B. From organ B, veins of organ B is initiated. Now, the veins of organ B is supposed to connect at vena cava, but it is not getting any chance to directly meet with vena cava. So, what it will be doing, it will be getting connected, it will be creating a connection, establishing a connection with organ A, and organ A is releasing its vein as veins B, veins A. Veins, this vein A, they are the actual vein coming from organ A, and this vein A is getting directly connected with vena cava. This is the ultimate goal. All the veins are getting connected. But for organ B, this is not the direct case. The veins which are coming out of organ B actually getting deposited or getting connected with organ A, and the deoxygenated blood is carried by the vein which are coming out from organ A, and then it is getting deposited into vena cava. So, here we can say that this organ B is using organ A as a port. The organ B is using organ A as a port to deposit deoxygenated blood instead of directly going and connecting with vena cavas. So, the similar things are happening into our body as well. See, the veins of our alimentary canal, the veins from the organs of our digestive system, the veins of stomach, spleen, pancreas, small intestine, large intestine, they are not directly coming out and getting connected with vena cava. Rather, they are coming out and getting connected inside liver, and from liver the hepatic veins are collecting all the deoxygenated blood of all these organs and delivering into the main vein or inferior vena cava. So, for stomach, spleen, pancreas, small intestine and colon, who is acting as a port? Liver is acting as a port. The veins of stomach, the veins of small intestine, large intestine, they are not getting connected directly with inferior vena cava. They are getting connected first with liver, and then hepatic veins, means veins from liver, are delivering the deoxygenated blood into the main vein. So, here liver is the port. Liver is the port for our digestive system, and that system is called as hepatic portal system. Hepatic means liver. Liver is acting as a port of veins for these organs associated with our digestive system. Now in frog you can observe one more kind of portal system. In frog you can observe one different kind of portal system, that is called renal portal system. So what happens there? So the frogs they have hind limbs. Hind limbs, the veins from hind limbs are not getting connected into the directly into the inferior vena cava. So the veins are utilizing kidney of frog as a port. So the veins coming out of kidney, that is called renal veins, they are carrying all the deoxygenated blood coming from the hind limb and depositing into the inferior vena cava. So for frog this system is called as renal portal system, because hind limb, the veins of hind limb are using kidney as a port to deposit deoxygenated blood towards inferior vena cava. Now we will talk about the regulation of cardiac activity. Regulation of cardiac activity, first of all we know that our heart is auto-regulated. Our heart is auto-regulated. We have sinoatrial node which can shoot 70 to 72 depolarization per minute, and that is called as autorhythmicity. That is why we said that our heart is auto-regulated. But certain neural and endocrine cues can alter the heart rate, can increase or decrease the heart rate, can moderate. So that is why we call it neural moderation and hormonal moderation. So how our nervous system can moderate our heart rate? See, if we talk about our ANF, autonomic nervous system, ANS, autonomic nervous system, it has two parts, parasympathetic division and sympathetic division. So basically, if your nervous system wants you to reduce your heart rate, it will make the parasympathetic nervous system dominant. So parasympathetic nervous system is responsible for reducing cardiac activity. And sympathetic nervous system, that is responsible for increasing cardiac activity. So that is how our nervous system can moderate. Similarly, there is one more nerve, that is 10th cranial nerve or vagus nerve. Remember, vagus nerve or 10th cranial nerve, that can decrease your heart rate and cardiac output. Similarly, how hormonal moderation can be done? There are few neurotransmitter or hormones. Basically, few hormones can act as neurotransmitter or can increase and decrease your heart rate, and they are like this. Like your adrenaline hormone, adrenaline, noradrenaline, even thyroid hormones. All these hormones can increase heart rate or cardiac activity. Adrenaline, noradrenaline and thyroid hormones can increase cardiac activities. Similarly, I will talk about one more neurotransmitter that can decrease your cardiac activity, that is acetylcholine. You will be studying about acetylcholine when you we are talking about neurotransmitter or neuromuscular junction. So, in the case of neuromuscular junction, this neurotransmitter acetylcholine will be exerting a positive effect, right, that can increase the muscle contraction. But for cardiac muscle, acetylcholine is having a negative effect that will be decreasing the cardiac output or heart rate. Remember that acetylcholine is like amazing kind of neurotransmitter. When it's acting upon your skeletal muscle, that increases muscle contraction. When the same acetylcholine neurotransmitter is acting on your cardiac muscle, that will be decreasing your cardiac output or heart rate. Okay. Now we will talk about a few disorders of our heart. So, the first disorder is hypertension, is simply called as high BP. Okay. So, what is the normal blood pressure or normal arterial pressure for us? We write it as 120 by 80, right? So, the 120 is the higher pressure. When your systemic artery is filled with blood, 120 millimeter mercury pressure, and when the arteries are relaxing, the lower range of pressure is 80 millimeter mercury pressure. So, if that increases, so if repeated checks of blood pressure of an individual is more than or nearby 140 by 90, upper limit crosses 140, lower limit crosses 90 or even higher, that condition is called as hypertension. And what are the probable reason of hypertension? Maybe atherosclerosis, deposition of cholesterol or fat bodies into your coronary arteries. Coronary artery disease can be one of the cause. Thickening of your chambers of your heart, thickening of your ventricles. Okay. Now, what can be the ill effect of hypertension? If you are suffering from chronic hypertension, then they can affect vital organs like brain and kidney. So, whenever you will be visiting a doctor and asking to check your heart or hypertension, heart condition, cardiac condition or renal condition, he or she, the doctor will be advising you to check for kidney functions as well. Next, CAD, coronary artery disease. So, this is also called as atherosclerosis. So, what happens in coronary artery disease? What is coronary artery? We know that heart supplies blood to each and every part of our body, but coronary artery is heart's own artery that is responsible for delivering oxygenated blood to heart's own muscle. Coronary artery is heart's own artery. So, if we draw a diagram, a basic diagram of our heart, so we know that the left ventricle, from left ventricle, one systemic artery is generated, that is called aorta, right? And that is responsible, the aorta and its branches, they are responsible for supplying oxygenated blood throughout the body. But what about heart's own muscle? Heart is having its own muscle called myocardium. See, branches, few branches of this aorta just coming out from here, right coronary artery and left coronary artery, they will be entering into the heart's own muscle and supplying oxygenated blood. So, if the lumen, if the space inside the coronary artery is getting filled with all these component, calcium, fat, cholesterol and fibrous tissue, that will block that will block blood flow, and that disease is called as atherosclerosis or coronary artery disease. Okay, do not confuse atherosclerosis with another disease called arteriosclerosis. Arteriosclerosis is about small arteries. When the wall of small arteries are stiffened or hardened due to the deposition of mainly calcium salts, fibers and cholesterol, that condition is called as arteriosclerosis. So, atherosclerosis is about large arteries like coronary artery, and arteriosclerosis is about smaller arteries. In atherosclerosis, the main villain is cholesterol and fat, right? Though there is deposition of calcium and fibrous tissue, but for arteriosclerosis the main villain is fiber and calcium, remember. Okay. Now another condition that is called angina pectoris. Now angina pectoris means chest pain. Pectoris means this pectoral region, this chest region is called as pectoral region. Angina means pain, or so the elaboration of this name is acute chest pain, sudden chest pain. Okay, now why it's happening? Because there is no enough oxygen supply to your cardiac muscle. If your heart muscles are not getting ample amount of oxygen supply or ample amount of blood, then gradually what will happen after that, your cardiac muscle will start dying, right? So as because this cardiac muscles are not getting oxygen, it will create acute chest pain, and that is more common in middle age to elderly. Now the condition like heart failure, heart attack and cardiac arrest. What is the difference between all three conditions? So heart failure, that means heart is not pumping blood effectively. Now behind heart failure there may be different reason. Maybe your maybe there is heart attack. Heart failure can be consequence of heart attack. Heart failure can be consequence of failure of SA node or AV node. Heart failure can be consequence of uneven thickening of your ventricles, right? So there are maybe many reasons behind this heart failure. Now what is heart attack? Heart attack means sudden damage of your cardiac muscles due to inadequate and improper supply of blood. So if for prolonged time your cardiac muscles are not getting supply of blood and oxygen, then it will start dying simply, and that when few cardiac muscles are dead, then that condition is called as heart attack. And generally in normal case the person will not be dying for heart attack. The another name of heart attack is called a myocardial infarction. Synonymous terminology for heart attack, myocardial infarction. When this myocardium means cardiac muscles are infarcted, or it can be called as myocardial ischemia. What is ischemia? The tissue of your heart is dying. Okay, but if there is massive heart attack, that can cause cardiac failure, even cardiac arrest. Now what is cardiac arrest? When due to any reason your heart has stopped beating, your heart is not responding, your SAN is dead, AVN is dead, so that will cause cardiac arrest. That means if you are observing the ECG, gradually the ECG will stop fluctuating and it will come into a straight line with a P sound. Okay, so we are done with this chapter. Now within 5 minutes we will be completing another chapter, that is excretory products and their elimination. So now the chapter will start, that is excretory products and their elimination. It's about the different modes of excretion, to know about different excreta, ammonia, urea, uric acid, which is the most toxic one and least toxic, which needs more water to be excreted out. What are the different forms of excretory structure found in animal kingdom, and obviously we'll talk about our kidney and our complete excretory system. So we know that animals accumulate ammonia, urea, uric acid, carbon dioxide, water and ions like sodium, potassium, chloride, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestion. So there are two probabilities our body can accumulate waste product after metabolic waste, after metabolism, and secondly, that is after excessive ingestion. Okay. So the excreta can be of two types, nitrogenous and non-nitrogenous. Excreta of our body can be of two types, nitrogenous wastes and non-nitrogenous wastes. What are nitrogenous wastes? They are ammonia, urea, uric acid. They are mainly ammonia, urea and uric acid. What are non-nitrogenous waste? Carbon dioxide, even excess, if you are drinking excess water, that's also excreta for you. Excess salt, if you are taking sodium ion, chloride ion, potassium ion, okay, excess sulfate, phosphate, everything considered as non-nitrogenous waste. Okay. Now these substances have to be removed totally or partially. This is very important line about our excretion. A few substances we have to remove totally, like we have to remove total amount of ammonia from our body. Complete ammonia is removed. This is very toxic for us, right? But when you are talking about partial removal, like urea is not removed 100%, like 20 to 30% of urea is recycled and kept inside our body in order to increase the osmolarity of fluid present into our kidney, right? To maintain osmotic balance, our body is keeping few part of urea inside our kidney. So when you are asked that name some excreta which is removed totally, then you can answer it's ammonia, and few excreta which is partially removed, the answer can be urea. Okay. Now here we can observe structure of ammonia, urea and uric acid. So ammonia is simply NH3, and another form of ammonia is produced, that is ammonium NH4 plus ion, that's much easier to dissolve in water. Now this is the structural formula of urea, and this is structural formula of uric acid. It's somehow it's looking like the structure is like purine because it's a derivative of purine, the nitrogenous base. And remember, all of them are nitrogen containing metabolic waste. Okay. Now we'll be differentiating in between ammonia, urea, uric acid. See, the animals will be excreting ammonia majorly, we'll be calling them ammonotelic. Animals who are excreting urea as a major excreta, we call them ureotelic, and obviously the animals who are adapted mainly for dry and terrestrial adaptation, they will be excreting out uric acid and they are called as uricotelic animals. Okay. Now let's compare that toxicity level. So the ammonia is highly toxic. Toxicity of urea is less than ammonia, and toxicity level is least in case of uric acid, so it's kind of less toxic. Now water requirement, see, mostly aquatic animals will be preferring ammonia to excrete out. So requirement of water is quite high, highest for ammonia. Requirement of water during the time of excretion for uric acid is the lowest, and for urea it's moderate. Like if I explain, if you want to excrete out 1 gram of ammonia, you will be needing near about 500 ml of water. If you want to excrete out 1 gram of urea, you will be needing 50 ml of water, and if you want to excrete out 1 gram of uric acid, you just will be needing only 5 ml of water. So where is 500 ml per gram? Where is 5 ml of water per gram? So that's why if one animal is trying to conserve water, then it will become uricotelic, and if the animal is aquatic living inside water, there is no harm in dehydration, then that animal will become ammonotelic. Now solubility, when you talk about solubility, solubility is very high for ammonia, specifically in ammonium format, and solubility is very low in case of uric acid, and solubility is quite good for urea. Now ammonia is excreted as either as ammonia directly as a gaseous format NH3, or as ammonium, that is NH4 plus. Urea excreted as liquid format with urine. Okay. Uric acid is excreted as solid format, as pellet or paste, and excreted as crystals. Okay. The animal which excrete ammonia, we call them ammonotelic. If you are excreting urea, you will be called as ureotelic. If you are excreting uric acid, you will be called as uricotelic. Role of kidney is negligible for ammonotelic organisms. Because the ammonia is excreted out with the help of body surface or gill surface, that's why there is no role of kidney, no role of kidneys are there. Okay. For ureotelic organism, like we are ureotelic, yes, we have role of kidney in course of excretion of urea. For uric acid, the role of kidney is here also limited, because uric acid is mainly released with the help of their alimentary canal along with the fecal matter. Okay. Now, if you are ammonotelic, the mode of your adaptation must be your aquatic organism. Aquatic organisms are ammonotelic. Terrestrial or aquatic, both kind of organism can become ureotelic. Mostly ureotelic organism are terrestrial in nature, but few aquatic organism like marine fishes, like sharks, they also excrete urea. Okay. Now uric acid, perfect for the animals who are inhabiting dry lands. So it's for terrestrial adaptation. Uricotelism is for terrestrial adaptation. Now examples of animal, the animals which will be exhibiting ammonotelism, they are aquatic arthropods. Most of the aquatic animals, fishes, tadpole larva of frog, etc. Which all animals will be performing ureotelism, will be excreting out urea? Mammals, then amphibians, and then marine fishes. Like sharks. Animals will be performing uricotelism. They are insects, terrestrial arthropods, then reptiles, aves. So the animals in the course of evolution who were adapted into dry land, all these reptiles, birds and insects, they have adapted their excretion aligning with uricotelism. Now see, for all the excretory structure, they are performing two kind of task. First, excretion, and second is osmoregulation. And you know that what is excretion? Excretion means removal of waste. What do you mean by osmoregulation? So osmoregulation means the balance of water and electrolyte inside our body, and to maintain a specific osmolarity or concentration of our body fluid. Like, what is the normal concentration of our body fluid? That is 300 milliosmol per liter. So if we can preserve proper amount of salt molecules like sodium, chloride, potassium, calcium into our plasma, into our body fluid, and proper volume of water, then only it will be maintained, and that is called osmoregulation. Maintenance of osmolarity of our body, simply maintenance of salt and water. Okay. Now let us talk about evolution of excretory structure. So the most primitive excretory structure you will be finding in animals, that is protonephridia. Advanced version of protonephridia is nephridia, and the highest order of, or the highest form of excretory structure, that is kidney, right? Protonephridia to nephridia, nephridia to kidney. So the highly advanced animal will be having kidneys, and the ancient animals you will be finding they have protonephridia. Okay. Now let us check excretory structure into animal kingdom, into different animals. So for simple invertebrates, we have simple tubular structure. For complex vertebrates, we have complex tubular structure. So protonephridia and nephridia, they are kind of simple tubular structure, and kidney, that is complex tubular structure. Now see, these groups of animals, platyhelminths. Now what's before platyhelminths? Porifera, Cnidaria, Ctenophora. They don't have any specialized excretory structure, but for platyhelminths, rotifers, annelids, some ancient annelid or primitive annelids, and one developed animal called as cephalochordata, like Amphioxus, Branchiostoma. All these animals, they have protonephridia. And the primary function of protonephridia is to do osmoregulation and excretion. Okay. For annelids, leech and earthworm, they have nephridia. Insects, six-legged arthropods, they have Malpighian tubules. Aquatic arthropods like crustaceans, they have green glands. Green glands in shrimp, crab, they have green glands or antennal glands. Arachnida like spider and scorpion, they have coxal glands for excretion. Vertebrates, we are vertebrate, we have kidney for respiration. Now let's talk about some animals who will be exhibiting dual mode of excretion. See, in case of earthworm, during the time of monsoon, in rainy season, they become amonotelic, but in dry season, dry summer, they will change their mode of excretion into ureotelism. Crocodile, while they are inside water, they are amonotelic in nature. While they are resting at the bank of the river, and when there is dry environment, they will become uricotelic. Ascaris, common roundworm, sometimes they perform amonotelism, and sometimes they will be performing ureotelism. So that's called dual nature of excretion in animals. Okay. Now let's jump into the human excretory system. Yes. So we know that our excretory system is consisting of one pair of kidneys, one pair of ureters, one urinary bladder acting as a temporary storage of urine, and urethra. Through urethral opening, the urine is eliminated out from our body. Okay. So now we can see here, we have two kidneys, right kidney here and left kidney, but these two kidneys are not situated into even position due to the positioning of liver into our abdominal cavity, right. Liver has pushed down the right kidney, so that's why the left kidney is little bit upward in compared to the position of right kidney. Remember this. Okay. Now, like the cap over the superior pole of kidney, though these structures are not associated with the excretory system, these are called as adrenal glands. These are part of our endocrine system, right. Now, from kidney, these pipes are coming down and merging with inside the urinary bladder. So these pipes, two pipes, each coming from right and left kidney, we call them ureter. One is left ureter and another is right ureter. They are merging into the urinary bladder. And the capacity of urinary bladder is near about 800 ml. So when your kidneys are producing urine through ureters, the urine will be coming and getting accumulated into urinary bladder, and whenever these muscles of urinary bladder is getting stretched, you will be getting urge for urination, and through urethral meatus you are passing your urine, even all of us, right. Okay. Now let's talk about the plumbing system or the vascular system of our excretory system, the vascular network connectivity of our excretion. So first, like other organs, our kidneys are also need supplies of oxygenated blood, right. So for that we need to give supply of arteries. So towards your kidney, towards our kidney, we can observe dorsal aorta is coming down, which is a branch of our systemic artery or aorta. Now, further sub branches of this dorsal aorta, which is going inside left and right kidney, we call them renal arteries. We call them renal arteries. The sub branches of dorsal aorta which is entering inside your kidneys, we call these as renal arteries. And the blood of these renal arteries, they are responsible for delivering oxygen into the tissue of kidney, and also the filtration of urea, uric acid, ammonia that is happening. The urea is fetched, the urea rich blood is fetched to the kidneys with the help of these renal arteries. So, this arterial blood, they are filled with oxygen, urea, ammonia and excreta. Okay. Now, after filtration, you need to get back the blood through your venous system. So, the veins which are collecting the filtered blood as well as deoxygenated blood from your kidneys, we call them renal veins, and from both the side, from left and right side, the renal veins are getting connected at the prime veins of our body, that is inferior vena cava. So, that is the blood vascular system. So, basically renal arteries are branches of dorsal aorta, renal veins are branches of inferior vena cava. Okay. Now, we will talk about kidney and its morphology. So, the type of kidney present into our body, that is metanephric kind of kidney. Metanephric is highly advanced kind of kidney. The prime location for our kidney, that is dorsal inner wall of our abdominal cavity. So, if this is the ventral side, this is the dorsal side, the kidney is present towards the dorsal side, and the location is mentioned as NCERT, present in between last thoracic vertebrae and third lumbar vertebrae. The color of kidney is reddish brown and the shape is like a kidney bean. Now, if we talk about dimension of our kidney, the length of kidney is in between 10 to 12 centimeter. The width of kidney is 5 to 7 centimeter, and the thickness of our kidney is 2 to 3 centimeter. So, these are the basic dimension of our kidney. And few more important thing, the weight of kidney. The weight of kidney is nearby in between 120 gram to 170 grams. So, at an average 150 grams. Okay. So, the outer covering, now we will talk about the outer covering of our kidney. So, that is called renal capsule, the outer covering, which is a kind of fibrous covering, right. Okay. Now, here in this diagram we can observe the positional information of our kidney. So, that is present towards the dorsal side, right. This is the dorsal side, and located in between 12 thoracic vertebrae and third lumbar vertebrae. Okay. Now, if we get inside our kidney, the kidney is subdivided into two zones. The outer zone is called as renal cortex. The outer zone is called as renal cortex, and the inner zone where you can find these renal pyramids, a pyramid kind of structure. This zone is called as this zone is called as medulla. This zone is called as renal medulla. Okay. Now, toward the interior side of your kidney, you will be finding a concave plane. This inner concave plane through which blood vessels and nerves are entering into your kidney, that is called as hilum, the inner concave plane of your kidney. This zone is called as hilum, and through which blood vessels and nerves are entering. Okay. So, this is a complete diagram of our kidney. So, the outermost covering, that is called capsule. This is cortex. This is medulla, and in medulla region you can observe this pyramidal structure called as renal pyramids. And one more interesting thing, tissue of cortex, by forming column like structure which is present in between these renal pyramids, that is called as columns of Bartini or renal columns. So, what do you mean by renal columns? When the tissue of cortex is present in between the medullary region, in between the medullary pyramids, so that cortical cells or cortical column is called as renal column or columns of Bartini. Right. Now, here we need to learn about path of urine. So, here these nephrons are present to the cortex region and the medullary region. We have cortical nephron, juxtamedullary nephron, and where urine is produced, right. So, urine is produced. After production by the nephrons, urine will be coming into collecting duct region. What is collecting duct? All the nephrons, many nephrons together, adjoining together to form a common drainage system through which urine is coming out, right. That portion is called as collecting duct. So, all the collecting ducts will be grouped together to form a wider channel, that is called as duct of Bellini. Now, this duct of Bellini through medullary pyramids. So, how many medullary pyramids are there? Total 8 to 12 medullary pyramids are there. Through medullary pyramids, that will be opened into a tubular part called major calyx. So, the number of major calyx is similar to the medullary pyramids. So, major calyx will be collecting the urine from each medullary pyramid, and they will be merged together to form minor calyx will be merged together to form major calyx. So, half of the number of the minor calyx is major calyx. So, total there are 8 to 12 minor calyx, and you can observe there are near about 4 to 5 major calyx. Now, all the major calyx will be merging together to form renal pelvis, and renal pelvis will get out through ureters and will be throwing or depositing the urine into urinary bladder. So, if we draw the diagram in like this manner, this is the kidney, this is the ureter, and this V shaped part, this is the renal pelvis. Now, the renal pelvis, that is forming several branches, and we call them major calyx. The subdivision of major calyx, we call them minor calyx. Now, this minor calyx, they are connected to renal pyramid. So it's like that from renal pyramid to minor calyx to major calyx to renal pelvis to ureter, and from ureter the urine will be fetched into the urinary bladder. That is the passage of urine. Now we'll be talking about nephron, and we know that nephrons are the structural and functional unit of kidneys, and in a single kidney you will be finding near about 1 million or 10 lakhs of functional nephrons are there, and there are two major parts of a nephron you can find, that is glomerulus and another is renal tubules. So the initial capillary, the tuft of blood capillary, that is called as glomerulus. Simply it's written, it said that tuft of blood capillaries means bunch of blood capillaries. This part is called glomerulus. So basically one branch of renal artery, that is called afferent arteriole, that is bringing blood inside the glomerulus, and here it's forming the bunch, right? This bunch is named glomerulus only, and when the blood is going out from glomerulus with the help of a narrow tube kind of structure, that is called efferent arteriole. So who is bringing blood to glomerulus? It's afferent arteriole. Who is taking out the blood from glomerulus? It's efferent arteriole. Now let's talk about renal tubules. Renal tubules have different parts, and there as here is given, starting with Bowman's capsule, the cup-like structure here that we call Bowman's capsule. Then we have a highly coiled part that is called proximal convoluted tubule, a U-shaped part called loop of Henle, another highly coiled part called distal convoluted tubule, and lastly it will be ending with collecting duct. Okay. Now let's check the detailed structure of a nephron. Okay. So this is the glomerulus, and this is the Bowman's capsule. Together, glomerulus and Bowman's capsule is called as Malpighian corpuscle or renal corpuscle. Do not confuse with this Malpighian corpuscle or renal corpuscle with Malpighian tubules. Malpighian corpuscle means in our kidney glomerulus plus Bowman's capsule. This zone is called as Malpighian corpuscle, and Malpighian tubules, they are the excretory structure of insects, right? Okay. And as you know that the wider blood vessel which is carrying blood towards the glomerulus, that's called afferent arteriole, and the narrower blood vessel which is taking the blood away from glomerulus, that's called efferent arteriole. Now this highly coiled tubular structure present near to this glomerulus and Bowman's capsule, that is called PCT or proximal convoluted tubule. Convolution means it's highly coiled, proximal means it's near to Bowman's capsule. Now this highly coiled tube which is far from this structure, that is called as DCT, distal convoluted tubule, because that is present into the distant zone. Now this hairpin loop like structure, that is called loop of Henle or Henle's loop, and this loop of Henle is having one descending arm through which the filtrate is coming downwards, and this ascending arm through which the filtrate is coming upwards. Now the DCT, distal convoluted tubule, of multiple nephrons are merged together to form CD, collecting duct. So if you are asked which part of renal tubule is not nephron's own part, that is collecting duct. Why it's so? Because collecting ducts doesn't belong to one single nephron. Multiple nephrons merge together to form one single collecting duct. So that was all about the renal tubular system. Now let's talk about the blood vascular system of your nephron. Okay. Now we know that this efferent arteriole, while it's progressing further, it's forming few other branches of blood capillaries, and these blood capillaries are surrounding the PCT, DCT and Henle's loop. So these capillaries are called as peritubular capillaries. So peritubular capillaries are nothing but branch of efferent arteriole which are surrounding the renal tubules, which are surrounding the PCT, DCT and Henle's loop. Okay. Now a special branch of peritubular capillary which is parallel, which is going parallel to the hairpin Henle's loop, that is called vasa recta. So the blood supply is like that. From renal artery we got afferent arteriole, then after afferent arteriole we got glomerulus, from glomerulus we got efferent arteriole, from efferent arteriole we got peritubular capillaries, from peritubular capillary we got vasa recta. Now one arm of vasa recta will be merging with renal vein. So that's the complete structure of a nephron. And if we talk about the tissue present inside nephron, see the PCT, proximal convoluted tubule, here you are gonna get brush border cuboidal epithelium. Brush border cuboidal epithelium, and that is the reason, it is the nature of tissue present into the proximal convoluted tubule. You will be getting to know that the highest rate of reabsorption is seen through the PCT due to the brush border nature of these cells, that is increasing the surface area of PCT. Okay. The thick segment of Henle's loop, both for ascending arm and descending arm, that has simple cuboidal epithelium, and the thin segment of Henle's loop, that is made up of simple squamous epithelium, flat and thin cells. The cell you are going to get inside the distal convoluted tubule, they are also simple cuboidal epithelium. Even inside the CD also you are going to get simple cuboidal epithelium. But the junction of at the junction of DCT and collecting duct, this is called the neck region. This is called the neck region. Here you are going to get a ciliated cuboidal epithelium. So this is all about the tissue present into different parts of our nephron. Okay. Now we know that we have two types of nephron present into our kidneys. So if this is the kidney, you know that kidney is divided into two zones, cortex and medulla. So the nephrons which are mostly present into the cortex zone, they are called as cortical nephrons, and the nephrons which are present near to medulla and present deep into the medullary region, they are called as juxtamedullary nephron. So this one is cortical nephron and this one is juxtamedullary nephron. Now population wise there are more cortical nephron, 80 to 85%, and there are less juxtamedullary nephron, only 15 to 20% of the nephrons are juxtamedullary. Okay. For cortical nephrons you can see here loop of Henle is very short But for juxtamedullary nephron, loop of Henle is very long and go deep into the medulla. So whenever you will be studying about the mechanism of concentration of urine, the counter current mechanism, that is more efficient in case of this juxtamedullary nephrons. Now let's talk about vasa recta. Vasa recta for cortical nephron is either absent or highly reduced. Now, as because you know the juxtamedullary nephrons, they are quite efficient to perform counter current mechanism, so they need developed vasa recta. So that is the difference between cortical nephrons and juxtamedullary nephrons. Do not ignore these points, the differences between loop of Henle and vasa recta. Okay, now we'll talk about urine formation. Now, urine formation takes place through three sequential stages. First stage is glomerular filtration. This is also called as glomerular ultrafiltration. Second stage is called as reabsorption, and third stage is called as secretion. Now, what is glomerular ultrafiltration? If you observe the structure of a nephron, the blood that is coming inside the glomerulus is getting filtered out here to form the filtrate. Urine formation will start from here. So this process is called as glomerular ultrafiltration, and almost things are there will be ultrafiltration, very minorly your body is filtering your blood, right? Only allowing the selective components to filter out from the blood capillaries through minute pore. That's why we call this process as ultrafiltration. What is reabsorption? Reabsorption means, if you check a renal tubule, if this is the renal tubule and this is the blood vessel or blood capillary, if materials are moving from renal tubule to your blood, then that process is called as reabsorption. That means your body is taking back the things from your nephron, right? So that is called reabsorption. And if your blood is releasing material directly into your nephron, into your renal tubule, then that process is called as secretion. So this step is called reabsorption, this step is called secretion. Again I am repeating it. If your body is taking back few of the components which was filtered earlier by the process of glomerular filtration, then that process is called as reabsorption. So during the time of reabsorption, materials are moving from your nephron to your blood. But during the time of secretion, your blood vessels or blood is directly releasing excretory products into your renal tubules or into your nephrons. Thank you, Shalini ma'am. Okay, now we'll talk about the detail of ultrafiltration. Now we'll talk about the method of ultrafiltration in detail. See, so what's happening? Through your afferent arteriole, per minute near about 1100 to 1200 of ml of blood is entering into your glomerular region and flowing and passing out through your efferent arteriole. So that is called as renal blood flow. So in a minute, near about 1200 ml of blood is passing through your glomerular region. And in the time being, the apparatus present here, that includes the pores of your glomerular epithelium, the basement membrane, and the podocyte layer and the inner layer of your Bowman's capsule. This trilayer, these 3 layers trapping few of the components from blood and sending into the Bowman's capsule, and that is the initiation of formation of your urine, and the liquid formed here is called as filtrate, Now the process of formation of filtrate from your blood, the process of formation of filtrate from your blood inside what? Inside your Bowman's capsule, that is called ultrafiltration. And while we are uttering the word ultrafiltration, that means we have to use minute pores. We have to use some minute pores for the passage of these filtrate components from blood to your Bowman's capsule. So this minute spores present in between the cells of this Bowman's capsule layer. This cell you can see here, they are called as podocyte cell. The cells you can see here in the inner wall of your Bowman's capsule, we call them as podocyte cell, as because if you elaborate, if you zoom out them, you'll be finding these cells are having foot-like structure. That's why we call them podocyte cell. Now there is gap in between this podocyte cell, and that gap is called as filtration slit or filtration pore. And check their diameter, only 24 to 40 nanometer is their diameter. So whenever the materials are crossing this laminar, crossing these layers, they have to cross such smaller pores to form the filtrate. That's why you are naming this as ultrafiltration. Now if you ask me, sir, what are the components flowing from blood to filtrate, forming filtrate? Obviously components of plasma is coming, except large proteins. Components of plasma are coming down towards the Bowman's capsule, except large protein, except proteins like fibrinogen, albumin or globulin, right? But all the salt and water balance is almost similar to plasma. The concentration of plasma into blood, that is 300 milliosmol, and the concentration of filtrate is similar, that is 300 milliosmol. So if you are asked what is the difference between plasma and filtrate, then you have to answer, though both the plasma or in the blood of a body and the filtrate having similar concentration of 300 milliosmol, this filtrate is not getting the large proteins from your plasma. So these large proteins are trapped here only inside the glomerular wall. Now even blood cells are also not coming. RBC, WBC are platelets. The glomerular slit, these glomerular pores are preventing the cells to come out within the filtrate region. So that's about the ultrafiltration process. Now let's move forward to the secretion, tubular secretion and tubular absorption. Now as I said that upon formation of filtrate, gradually the filtrate is coming or progressing through the PCT. Now what will happen? Gradually your body will be finding, during the time of ultrafiltration, I have filtered out too much of components. Now this filtrate is having a lot of volume of water, salt as well as nutrients. Now if I release them out in the form of urine, eventually our body will lose water, dehydrate, our body will lose some really essential nutrient for us, and maybe we can die. It's just like that phenomena. Suppose there is Diwali, there is a festival in your room, in your home, at your house. Now suddenly your mom entered into your room and saw your room is nothing but a mess, total mess. So suddenly she will get angry and she will throw all the components, all the things, including you, including your NCERT, including your books, including your mobile phone, mobile charger, everything into the trash upon anger. Now suddenly after that she will feel that, yes, I have thrown out the NCERT books. Now she or he will be needing that book for the preparation of a NEET. So again what she will do, she will get back those NCERT books and obviously you again back to your room. So that is the meaning of a real reabsorption. After filtration, your body finds that it has done some mistake by filtering too much of substance into the filtrate. That's why your body will sending them back again to your bloodstream. So if you check your proximal convoluted tubule PCT, as I said, the highest reabsorption is seen here. And what are the components are reabsorbed through your PCT? 100% of nutrients, that means all the glucose molecules present into filtrate, all the amino acid molecules present into the filtrate will be reabsorbed from your nephron, from the PCT. Near about 70 to 80% of salt and water is also reabsorbed from your PCT. Bicarbonate ion HCO3 minus and potassium K plus ions also reabsorbed through your PCT. Right. Now what are the things can be secreted from blood to your PCT? So they are the harmful components like H plus ion, the protons, very naughty ions inside your body, H plus ions, ammonia NH3, again highly toxic for your blood circulation, for your tissue. So selective secretion of H plus and NH3 will be there through your PCT region. Now let's come into the descending arm of loop of Henle. So throughout the loop of Henle there will be no secretion, there will be only reabsorption. Descending arm is known for reabsorption of water molecules, and you need to remember that this descending arm, this DA, descending arm of loop of Henle, is only permeable to water molecules, it's impermeable to electrolyte. That means no sodium, potassium can transport from here, only water can be transported out, water can be reabsorbed from your descending arm. Now let's check your ascending arm. It's opposite, absolutely opposite. Throughout your ascending arm you cannot transport a single molecule of water. So this ascending arm, this AA ascending arm, is only permeable to electrolytes like sodium chloride and potassium, but impermeable to water. So what are the things can be reabsorbed through your ascending arm? Sodium ion and chloride ion. And one more important thing about the reabsorption through the ascending arm, the reabsorption rate is lowest for your ascending arm, the reabsorption rate is highest for your proximal convoluted tubule, and that also reabsorption of Na plus and Cl minus will be happening passively through the thin portion of ascending arm, and through the thick portion of ascending arm the same reabsorption of Na plus Cl minus will be happening actively. Yes, Mr. Umar. Okay. Now gradually, when the filtrate is entering into the DCT, distal convoluted tubule, there will be conditional reabsorption of salt and water. Why I am saying conditional? It's not always through your DCT salt, water and HCO3 minus is reabsorbed. It's under a condition, and the condition is if it's getting signal from the hormone called aldosterone. NEET aspirant, everything you are gonna get to like you will be getting to know. For that you have to follow our PW app, You just have to subscribe over there, and you will be getting each and every aspect of your queries, right? Okay. So what I was explaining, when the filtrate is entering into the DCT, there will be conditional reabsorption of salt and water. Why I am saying conditional reabsorption? Suppose you have ample amount of, you are drinking good amount of water, you don't need to reserve water into your body, then there will be no reabsorption through your DCT. But you are fasting, even you are not drinking water, your body is dehydrating, then you will get signal of one hormone called aldosterone, and upon the influence of aldosterone, the DCT will be performing reabsorption of salt and water. Now, what are the components can be secreted from your blood directly into the DCT? Obviously, ammonia, uh, potassium ion, and the naughty, very monkey kind of H plus ions. They are very harmful for you. That will be making your body acidic. That's why you know that nowadays you can observe a few people, they are suggesting drink alkaline water. Okay, no, actually this is not English batch. For us, that is Arjuna English. For English, there will be mixture of Hindi as well as English. But here we'll be studying and we'll be teaching completely in English, right? Okay. So if you are done with DCT, gradually the filter has entered into collecting duct region. Now listen very carefully. For the first time, when the filtrate has entered into collecting duct, now this liquid can be called as urine. So the question can be asked, when upon filtration of the blood, while the liquid is traveling through the tubular part of nephron, when for the first time we can name that liquid as urine. Is it inside PCT? No, it's not urine, it's just filtrate. Inside loop of Henle, it's just filtrate, it's not urine. Inside DCT, again, it's just filtrate, it's not urine. From DCT, when the filtrate is entering into the collecting duct for the first time, then only we call it urine. And again, let us check what are the things, what are the components can be reabsorbed through collecting duct. Yes, if it's under influence of antidiuretic hormone ADH, there will be reabsorption of a lot of water through your collecting duct. There will be reabsorption of lot of urea. So till now you may know that ADH, antidiuretic hormone or vasopressin hormone, is responsible for reabsorption of water molecule through collecting duct. But today you know one more thing, ADH is not only responsible for reabsorption of water through collecting duct, ADH is also responsible for reabsorption of urea through distal part of collecting duct. So that's why in the beginning of the chapter they said that excretion is the process of removal of excretory product or metabolic waste completely or partially. So that's why we are releasing suppose 70% of urea along with urine, but your body is reabsorbing, your body is keeping 20 to 30% of urea from here. And what are the components can be discharged, can be secreted into collecting duct directly? They are potassium ions and H plus ions. So we are done with tubular secretion. Now we will talk about mechanism of concentration of filtrate. See, suppose you went to desert for a vacation, but suddenly some natural calamity came and you lost your pathway. Now you lost your map, you lost your phone, you don't have any water to drink. So in that condition, what will happen? You will start dehydrating. But to counter that, to cope up with that condition, your body will activate one mechanism called counter current mechanism, that is the mechanism of concentration of filtrate. That means while our blood is having a concentration of 300 milliosmol, but our body will try to release the urine which is having 4 times more concentration, that is of 1200 milliosmol. A concentrated urine means your body is trying to reabsorb more and more water. Now, if this mechanism fails, then what will happen? Whenever you are dehydrating, you don't have anything, you have no one inside your body to save water, to conserve water, and that will cause more dehydration, and that may cause sudden death even, or failure of your kidney, failure of your brain. That's why this mechanism is very much important, and only we mammals are having this ability to produce concentrated urine. What is the definition of concentrated urine here? It is the 4 times more concentrated than our normal body fluid. What is the concentration of our normal body fluid? 300 milliosmol per liter. What is the concentrated of urine we can produce? 1200 milliosmol per liter. So it's just like that. So what do you mean by 300 milliosmol at 1200 milliosmol? Suppose you have taken 1 liter of water, and inside that 1 liter of water you have 300 millions of salt particles, and you have another liter of water, and inside that you have 1200 millions of salt particles. So which one will feel more salty? The liter of water which is having 1200 millions of salt particle inside that. So that is the result, that is the difference between our concentrated urine and our normal blood or normal filtrate. So how we produce that? Produce 4 times more concentrated urine in compared to our body fluid, or in compared to our blood. So basically the mechanism is also called as counter current mechanism, and the parts of your nephron which are involved to form this 4 times concentrated urine by you, that is the Henle's loop or loop of Henle of your nephron, and obviously vasa recta, right? Okay. Now what is the nature of a healthy kidney? See, corticomedullary concentration gradient is the nature of healthy kidney. What do you mean by corticomedullary concentration gradient? If this is your kidney, and this is cortex, this is medulla. Now like a pizza, if you take one chunk of your kidney and you observe under microscope, what really you can observe? Towards the medulla the concentration will be 1200 milliosmol, and towards the cortex the concentration will be 300 milliosmol. So from cortex to medulla the concentration is increasing. So that is the nature of healthy kidney. If you are finding one kidney, if you are finding kidney of one person who is suffering from kidney failure, then what you can observe? Then you will be observing the cortex region having 300 milliosmol concentration, the medulla region also having 300 milliosmol concentration, right? So this is the nature of a failed kidney. This is the nature of a good or healthy kidney. So that is called corticomedullary concentration gradient. And again I am repeating, who are forming that? It's Henle's loop and vasa recta. Why it's necessary? It's necessary because easy to easy passage of water through your collecting duct. Why it's so? Let me clarify. Suppose you got a concentration gradient increasing from cortex towards your medulla. See, this is the cortical region, the concentration is 300 milliosmol, and towards your medulla the concentration is increasing 300, 400, 500, 600, 700, 800, 900, 1100, and near to the medullary inner medulla the concentration is 1200 milliosmol. So whenever the filtrate with 300 milliosmol will enter into the collecting duct region, what it will observe, the surrounding media is hypertonic. Surrounding media is hypertonic. If your concentration is 300 and surrounding media has concentration of 400, then what will do? You will start releasing water to match. You will start releasing water. If water is released, that means you are gaining salt. If you know about, if you have idea about a solution is made up of two components, one solvent and one solute, right? One solvent and one solute. So if you want to increase the concentration of that solution, what you can do? Either you can remove solvent. If solvent is removed, the ratio of solute will be increasing. So the overall concentration of the solution will be increased. The same thing is happening here. The urine which is coming down through your collecting duct will try to match the concentration with the surrounding kidney tissue or surrounding interstitial space, right? So it will start losing water again and again. It will reabsorb too much amount of water, so your body will hold that excess water and ultimately you will be excreting out hyperosmolar, hypertonic urine with 1200 milliosmol. So that was the whole and soul motto behind making this corticomedullary concentration gradient. And if you are asked with the question, the gradient is caused by which components? Which are the components who are making the medullary zone hypertonic or hyperosmolar? The answer will be salt, NaCl and urea. Salt and urea is responsible for making the medullary zone of your kidney hyperosmolar or hypertonic in compared to your cortex, right? So that was about the beginning part of your counter current mechanism. Now why exactly we are calling this mechanism as counter current? So this is your loop of Henle, this is descending arm and this is ascending arm. Okay, so through the descending arm the liquid is flowing downwards and through the ascending arm the liquid is flowing upwards. So if you observe the flow of filtrate through your loop of Henle, descending arm versus ascending arm, the direction will be in opposite directional manner. So if two fluids are flowing into opposite direction, then that is called as counter current mechanism. Similarly, we have a parallel blood capillary along with this Henle's loop, and that is called as vasa recta. That is called as vasa recta. So inside vasa recta, what is flowing? Inside vasa recta, blood is flowing. So again, if you check the flow of blood, blood is entering from here and exiting from here. So this is the descending limb of vasa recta and this is the ascending arm of vasa recta. So if you study the blood flow, descending arm blood is flowing down, ascending arm blood is flowing up. So for blood also is bidirectional, opposite directional flow. So for blood also it's a counter current, for filtrate also this is counter current. So that's why the mechanism is called as counter current mechanism. Okay. Now if we talk about regulation of kidney function, see, kidney functions are regulated by few hormones called ADH, antidiuretic hormone, a complete passage called RAS, renin, angiotensin, aldosterone system. Kidney function can be regulated by your atrial natriuretic factor. Okay. So there are many mechanisms to control your kidney function. There is a muscular mechanism called as myogenic mechanism to control your kidney function. Now, how ADH will be controlling your kidney function? See, there is a term called diuresis. What do you mean by diuresis? Excess loss of fluid from your body. Excess loss of fluid from your body is called diuresis, right? So whenever your body will start dehydrating, that means you are losing water, too much amount of water through your urination. So what will happen? If water is going out from your body, there will be increased concentration of salt into your blood. If your body is dehydrating, your blood will gradually become salty or saline. Now that will give a signal to osmoreceptors present into your brain, particularly into the hypothalamus region. Your brain is always checking for the salinity of your blood. If there is less water in your blood will become saline, and that is sensed by your osmoreceptor present into hypothalamus, and as a response your hypothalamus will release this hormone called antidiuretic hormone. Now antidiuretic hormone, when released, it is produced by hypothalamus but released through the posterior pituitary, right? So this antidiuretic hormone will give a signal to the collecting duct of your nephron, and your collecting duct will allow reabsorption of ample amount of water. And if your body, your kidney is reabsorbing water, that means the excess water will be gained by your blood. Less water is going through your urine and more water is absorbed into your blood. That is called reabsorption of water through your collecting duct of nephron. So if your blood is gaining water, your blood volume will be increased. If blood volume is increased, your blood pressure will be restored. And your urination will also be restored. Your body fluid balance is also restored, and that's already saved you from dehydration and dying. Now one more thing, this antidiuretic hormone, that's got another name. Okay, this antidiuretic hormone that got another name, that is vasopressin. Now why it's called as antidiuretic hormone? Because it can prevent diuresis. Excess loss of water is prevented when you are reabsorbing water through your nephron. That's why it's called antidiuretic hormone. Another name of antidiuretic hormone is called vasopressin. Another name of ADH is called vasopressin. So the name is telling its function. Vaso means vessel, pressin means to press. So antidiuretic hormone is having capability to press a blood vessel to increase the blood pressure. So whenever you will be dehydrating, what will happen? Your blood pressure tendency will be there, your blood pressure will fall. So that ADH can increase your blood pressure by pressing some specific blood vessels of your body. That's why it's called as antidiuretic hormone as well as vasopressin hormone. Now the next phenomena is RAS mechanism. What is the full form of RAS? That is renin angiotensin aldosterone system. So it's a cascade, it's a chain system. One hormone is activating another hormone. So let's see, when our body will activate this RAS system, I say it RAS Leela of your body. Again, you are facing a low BP, low blood pressure is there. So if there is low blood pressure, there will be low glomerular filtration rate. If there is low glomerular filtration rate, then your body will be releasing too much of water through your urine, that is called hypotonic urine. Now, if you don't know what is GFR, glomerular filtration rate, we were talking about glomerular ultrafiltration, right? So, if you talk about this Bowman's capsule and this glomerulus, in a minute 125 ml of filtrate it is produced. In a minute, your kidney is producing 125 ml of filtrate, that is called glomerular filtration rate. Now suppose if your body is facing low BP, your body is facing low blood pressure, so less blood is gonna flow through this circuit, and suppose instead of 125 ml, your body is producing now 90 ml filtrate. So that is called lowered or low down glomerular filtration rate. If less glomerular filtrate is produced, then what will happen? Your body will tend to release more water, and again that will cause more dehydration and that may kill you. So, when your body understands this something bad is happening inside your body, that will send a signal to a special apparatus, a special machine present into your nephron, that is called juxtaglomerular apparatus, which is present in between the afferent arteriole and distal convoluted tubule. And this juxtaglomerular apparatus is having special cell called juxtaglomerular cell or JG cell. So whenever there is low BP and low GFR in your body, your JG cell, juxtaglomerular cells, will be sensing that, and they will release a special hormone called as renin. Now this is the beginning to replenish your blood pressure and your GFR and your kidney function. So who is secreting renin? JG cell or juxtaglomerular cells of your JG apparatus, which is present into your nephron. Now this renin will convert a protein called angiotensinogen into angiotensin 1. Now who is producing angiotensinogen? Your liver is producing angiotensinogen, which is a switch off form of protein. Now who is required to switch on that protein? We need renin to switch on the protein released by liver called as angiotensinogen. So under the influence of renin, angiotensinogen is converted into angiotensin 1, and now this angiotensin 1 will be converted by another enzyme called angiotensin converting enzyme, which is released from the capillaries of your lung, present inside your lung surrounding the alveoli. That enzyme will convert angiotensin 1 into angiotensin 2. Now this angiotensin 2 is a strong vasoconstrictor. Whenever your blood vessel is constricted, your BP will increase. If your BP is increased, your glomerular filtration rate gonna increase, and your body will start taking more water. Your body will start reabsorbing more water and releasing less water through the process of urination. So that is the importance of this RAS mechanism. Now let's check what are the different functions of this angiotensin 2. Being a very strong, potent vasoconstrictor, is a strong vasoconstrictor, it will act on adrenal cortex first. It will act on your adrenal gland and adrenal cortex and will stimulate release of a special hormone called aldosterone, and already I was discussing this. Aldosterone can act on your distal convoluted tubule, and if aldosterone signal is coming to your distal convoluted tubule, there will be conditional reabsorption of sodium, water and secretion of potassium and H plus ion. And see what will happen if your DCT is reabsorbing salt and water, that excess water will enter into your blood and that will restore the blood volume, and if blood volume is increased, blood pressure will also be increasing. Now again the angiotensin 2 will go to your heart, and somehow that can increase your heart rate as well. If heart rate is increased, your blood pressure will also be increasing. Even the angiotensin 2 can stimulate your brain to release more and more antidiuretic hormone, and already you know that when antidiuretic hormone is produced in more amount, then your nephrons will be reabsorbing more water and secreting concentrated urine. And that may also, this angiotensin 2 will stimulate your thirst receptor. You will feel thirsty, you will drink ample amount of water, and if you are drinking more and more water, that water will be reabsorbed by your blood, and once you are drinking water, your blood volume increases and your blood pressure is restored. So that's how this angiotensin 2 can increase your BP and increase your blood pressure and saving you from kidney failure and dehydration. Okay, now I was talking about another hormone called ANF, atrial natriuretic factor. Now what is the function of ANF? Just opposite to the RAS and ADH. ADH and RAS, they are responsible for increasing BP and increasing GFR, but opposite means this atrial natriuretic factor, that is actually released by your heart, it is a hormone of your heart. Remember, atrial natriuretic factor, this is released by your atria of heart, right? And that can decrease your blood pressure. Whenever your body is facing high BP, high GFR, then your heart will be releasing ANF that helps you to decrease your blood pressure and decrease your glomerular filtration rate. So just opposite function to the RAS. Now the process of micturition. What is micturition? Micturition is the process of release of urine from your urinary bladder, right? So basically the neural mechanisms causing it called the micturition reflex. To micturate, you need to create a neural mechanism, and that is called as micturition reflex. And you know that one adult human can excrete near about 1 to 1.5 liters of urine per day, and the urine formed a light yellow colored watery fluid which is slightly acidic. See, the pH of urine is 6.0, that is acidic in nature and has a characteristics odor. Why urine is yellowish in color? Because urine is having a special kind of bilirubin called as urobilin, a pigment of our body released by your liver. Why the urine is having its characteristic smell or characteristics odor? Because of presence of some volatile components present into this, aromatic volatile components called as urinodes, remember. Okay, and while you are urinating, at an average 20 to 30 grams of urea is excreted out per day. So with your urination, you are excreting out 20 to 30 grams of urea with your urine. Okay. Now remember, various conditions can affect the characteristics of urine. So analysis of urine helps in clinical diagnosis of many metabolic disorders, like if there is excess glucose, excess ketone bodies present into your urine, that is called glycosuria and ketonuria, that denotes maybe you are suffering from diabetes mellitus or blood sugar, right? So that was all. Okay. Now, apart from our kidneys, lungs, liver, saliva, sweat gland and sebaceous glands. Now, apart from your kidney, your lungs can also excrete carbon dioxide and water while you are exhaling, right? So, these are also excreta for you. Your liver can excrete out the degraded vitamins and other components. Liver can excrete out bilirubin, biliverdin, right? Your saliva can excrete out good amount of water, salt as well as urea. Sweat gland or sudoriferous gland, when you are sweating, that is excreting out lactic acid, water, sodium chloride as well as a little amount of urea as well. Sebaceous gland, the oil secretion by your skin, that is making your skin oily sometime. So that oil is called as sebum, which are nothing but secretion of the sebaceous glands, and those oil are containing lipid molecules like hydrocarbons and other lipids. Okay. Now, it's a few disorders of our excretory system. First condition is called uremia. See, when your kidney is failed, when your kidney is failed, then urea won't be excreted out. Then there will be excess accumulation of urea inside your body. So, this high urea concentration into your blood is called as uremia. Another condition is called glomerulonephritis. That means infection of your glomerulus causing inflammation, that is called glomerulonephritis. Whenever you will be finding the name of a disease is ending with itis, that means inflammation. Now, another disease you might have heard about, that is renal calculi, that means kidney stone. Renal means kidney, calculi means stone. Due to the deposition of insoluble stones of calcium oxalate, you need to remember the name, that is deposition of calcium oxalate into your kidney, that is the causative agent for your kidney stone, and ultimately renal failure or kidney failure. See, if you are diagnosed that your 70% of the nephrons are non-functional, already damaged, unable to perform filtration, then doctor will be suggesting you to perform dialysis. Now, what is dialysis? With the help of a machine called hemodialyzer, your blood is drawn out from your blood capillary, arterial blood is drawn out, and with the help of this dialyzer machine, all the nitrogenous waste products are filtered out, and the filtered blood is given back into your venous blood. So that is called dialysis, but remember, dialysis is temporary solution, right? You cannot perform dialysis for throughout the life, even it's very painful mechanism, right? So a permanent solution is there, that is kidney transplantation. Now, for kidney transplantation, you will be needing a donor, and you have to match the blood group and the tissue type with the donor. That's why it's suggested that if the donor is your close relative, then there is maximum chance of successful kidney transplantation. And most of the time during the kidney transplantation, you think that if my two kidneys are not working properly, if I'll go for operation, doctor will take out those two bad kidneys or failed kidneys, and instead of them they will be installing a new one. It's not like that. The two failed kidneys always remains into your into your body, one third kidney will be installed in some convenient zone into your abdomen, or that posterior abdominal or peritoneal retroperitoneal cavity, right. So the person or the patient who has recently undergone kidney transplantation, probability is there he or she is having 3 kidneys into his or her body. Okay. So that's all. So another question is there. So renal papilla is a part of minor calyx, pelvis, pyramid or major calyx. Can you give the answer of this particular question into the comment section? Renal papilla is a part of minor calyx, pelvis, pyramid or major calyx. So this is cortex, this is medullary pyramids, these parts, the end part, this conic part of this medullary pyramids are called as renal papilla. Yes, so the answer will be renal pyramid. Now, can you answer this particular question? Assertion is saying that nephrons are of two types, cortical and juxtamedullary, based on their relative position in cortex and medulla. Reason is saying that juxtamedullary nephrons have short loop of Henle, whereas cortical nephrons have long loop of Henle. Can you answer this particular question? Okay, so here assertion is the true, but reason juxtamedullary nephrons have long loop of Henle. So reason is false. So here answer will be option 3. Okay. Uh, so with this, I'm ending the class for today. Again, we'll meet with another chapter. Probably it's tomorrow, the same time. And for today, it's good night and bye-bye.